biomarkers. I tried to activate the camera, but I think it's working ou non? Vous voyez quelque chose? Ils sont derrière. Hein? No? We tried to activate the camera, but apparently it don't work. Ah. Je sais comment activer la caméra, je pense que il faut le faire. Les gens de bien, c'est pas le fond de bien, normalement, elle est censée suivre l'intervenant en plus. I'm going to switch it off and to try again. Okay, people that are online, sorry for that because we don't have the camera. Dites-moi ceux qui ont l'habitude de faire ça sur PC. Pourquoi je vais un truc. Bon, sinon, ça va passer, mais bon, je mets la pression pour vous mettre la caméra, il ne faut pas la voir nous. Caméra, recherche des caméras. Ouais, je ne sais pas si elle est allumée. Il y a un truc? Ah, ouais, hier c'était quelle couleur? Bleu? C'était bleu? Oh. C'est parce que je peux la bouger. Ah non, je suis. Ouais, je peux la commander, je peux la bouger, ça c'est chouette. Ça sert à quoi? Vous voyez, ça lui, là, il fait tout seul. Sur tous les boutons. Aucune caméra pour les. Bon, c'est pas grave, on va s'en passer aujourd'hui. De toute façon, vous avez vu nos têtes depuis le début. Mais bon.
L'université, ouais.
Et je vais démarrer aujourd'hui, peut-être. Non. Okay, so the rule is if you the number is stick, we can connect. Sorry, but we don't have the camera. So for Pankaj, Luna, Antoine, and Yamina, we are sorry, but we cannot activate the camera. We don't know exactly what it will work, but do you hear us correctly? Yes? Okay. Anton, I don't know if you are talking with us, but no, you are not talking with us. Okay.
Yes, I'm listening. Sorry.
Okay. And now, why it doesn't work on the screen? Who is in charge of the presentation of the review? You can come, please. So today we're going to talk about one thing that you may perfectly know, but this is something that maybe you can heard about. We are going to talk about senescence. So first of all, we are going to hear a group that is that will present something around all the different characteristics of senescence, what it is exactly, how we can evaluate the senescence, etc. And then we will switch after with two publications, two papers, demonstrating, of course, the impact of senescence on global aging and the possibility to modify this senescence and to compare assessing senescence as a biomarker failure. Here we go. Is it share? Yeah. Can you share it? Voilà. Zoom. Tu connais, du coup. Tu prends la caméra.
Peut-être qu'il faudrait essayer de le faire.
C'est pas le caméra?
Ouais.
Elle est pas bleue.
Mais je pense qu'il faut appuyer sur le bouton.
Lequel? De la télécommande?
Ouais.
Allez, tu peux me passer. Mais il devient bleu.
Il n'y a pas un bouton allumé dessus, genre jamais.
Je peux monter si vous voulez, je peux me utiliser un peu marre aussi. Je vois pas de bouton. Les gens de Care, là, Care people, you are used to. Non? C'est bon pour vous les filles? On peut commencer? Okay, we go. We are not going to make a very long workshop today because I know that after that you are one day free. So here we go. And can we stop you if you have questions, or do you prefer that we start with we wait for the end? Okay, so better to ask the question at the end. It's okay? Here we go.
Today we are going to talk about the article titled Senescence in Health and Disease. Cellular senescence is defined as a durable cell cycle arrest that play a crucial role in preventing cancer in mammals. This phenomenon was initially undervalued and faced the challenges in scientific acceptance. Many doubts persisted regarding its biological relevance beyond cell culture. Despite these reservations, recent research has established that senescence play a key role in various physiological and pathological states. True question remain about its heterogeneity and its definition and its heterogeneity of the senescent cells in vivo. The major challenge is the senescence in senescence research, therefore remains the absence of available the magic marker to identify senescent cells in vivo, representing a significant obstacle in the field of study. The characteristics of cellular senescence include the increased expression of the product of the CGP2R locus, changes in cellular morphology in vitro, such as an increased cell spreading, increased activity of SA beta galactosidase, the presence of short telomere, the presence of DNA scares, the presence of senescence associated heterochromatin foci, the production of SA-SP factors, the increase of NF-κB signaling, and the loss of lamin B1. It's important to note that there are different types of cell cycle arrest, including senescence, quiescence, terminal differentiation, and exhaustion, but I don't speak about these chip cells. Cellular senescence begins when a cell experiences DNA damage or other chips of cellular stress, such as telomere dysfunction or oxidative stress. Initially, in senescence leads to a temporary cell cycle arrest, typically shows the activation of the P53 protein and the expression of the P21 mediator. These cells then start to express and secrete senescence-associated secretory phenotype, SA-SP factors. If the stress is short-lived, the cell has the opportunity to resume its cycle. However, for senescence become irreversible, prolonged exposure to stimuli is necessary, resulting in the activation of the CGKN2R locus and the durable growth arrest. With age, the accumulation of senescence increases exponentially, reflecting the multiplication in aging tissue. This accumulation results either from an increase in the production of senescent cells or a decrease in the elimination of these cells, a function normally carried out by the immune system. As aging progresses, the ability to clear senescent cells likely decreases due to immune system.
So we're going to talk about the beneficial roles of senescent cells. The author shows 5 beneficial key roles, which are organogenesis, wound healing, tumor suppression, host immunity, and atherosclerosis. Firstly, in organogenesis, senescent cells have a beneficial role. Organogenesis is the development of organs. Actually, these cells are quite unique. Unlike senescent cells, these cells are equal-replicative cells, which means that they do not multiply. Also, they present an activity of SA beta-galactosidase significantly, but this time it is not associated with DNA damage. Also, these senescent cells are independent of the P53 and the P16 inquiry pathways. And also, these cells are mediated by paracrine signaling by the expression of P21. Next, in wound healing, studies have shown that the activation of the promoter of P16 infrared can help a wound to heal. Actually, there is studies shown also that it's not the activation of the promoter that helps directly a wound heal, but a special feature of senescent, like senescent cells. Actually, when the wound is made, different mechanisms take place, such as the activation of NF-kappa B signaling, which is inflammation, and the secretion of cytokines. The author shows that when they clear the organism of senescent cells, the wound healing process is delayed. Also, there are special features like the secretion of PDGF-AA by fibroblasts and endothelial cells that help the tissue remodeling the tissue. Recently, studies have shown that these senescent cells are macrophages, which accumulate with aging. In terms of tumor suppression, senescent cells act like a safeguard against cancer. The loss of P16 and P53 functions is the common genetic event in human cancer. Actually, the senescent cells send out local and persistent signals of oncogenic stress, which facilitates the self-immune surveillance. When we talk about host immunity, it's a little bit the same. The senescent cells act like a barrier against infections. They recruit innate immune cells to limit the spread of the viral infections. In atherosclerosis, the senescent cells have a beneficial role. Studies say that when the locus CDK N2A2B is expressed, that plays a role in reducing the risk of development of atherosclerosis disease. Actually, it limits the proliferation of macrophages and limits, by consequence, the blood growth.
Okay, so senescent cells play a complex role in biology of hanging organism. They have a lot of beneficial effect, but they have also a negative effect on health, and especially when they accumulate. For example, senescent cells secrete a set of pro-inflammatory molecules known as SASP, and this secretion contributes to a chronic pro-inflammatory environment by contaminating healthy cells with senescent cells, and this leads to tissue dysfunction, both locally with paracrine secretion and endocrine-wide via the blood. However, this figure also shows other damaging effects of these senescent cells on different tissues. For example, this pro-inflammatory environment can create consequences like atherosclerosis that can lead to develop other disease. And it's also how the SATs contribute to age-related disease while, as previously stated, their anti-proliferative aspect is itself beneficial. Concerning diabetes, some studies have shown that characteristic of senescent cell is, for example, expressing anti-proliferative markers such as P16, and this molecule is highly expressed in insulin-producing senescent cells of the pancreas, namely beta cells. And when these cells overexpress P16, they enter in senescence, so they still remain alive, but stop dividing, and these cells cannot be replaced because replication is blocked. So they accumulate, and in general, these senescent cells produce less insulin, as senescence leads to a reduction in cellular function. But some studies have shown a paradox. Some senescent cells can produce more insulin per cell than non-senescent ones. And consequently, senescent cells may be responsible of predisposition to develop diabetes by an hyperinsulinemian effect that can lead to tissue resistant insulin. Finally, concerning fatigue, some findings suggest that senescent cells play a role in fatigue. So a study was conducted to examine the role of senescent cells in chemotherapy-induced fatigue. And for that, they induce senescence in urine tissue with chemotherapeutic agents. And after eliminating these senescent cells, some negative effects of this chemotherapy were attenuated. And the senescent cells also contribute to other age-related phenotypes such as sarcopenia or hair graying, also known as amylase.
We will now discuss the therapeutic strategy mentioned in the article. First, estimating the risk age-associated disease. An effective theory of aging could enable the measurement of the organism molecular age by analyzing the quantity of senescent cells, offering clinical prediction on the disease susceptibility and reproductive capacity, while validated in the animal models. Measuring human senescence difficult. Current marker includes cytokine, telomere dysfunction, and more recently P16 expression. For example, in kidney transplantation, high P16 expression in donor kidney better predict graft longevity than chronological age. P16 expression in blood T cells accelerated by pro-aging behavior or intensive treatment is a strong indicator of senescence. A marker like DNA methylation are also promising, though it remains unclear if they provide complementary information on physiological age.
Now, concerning clearing senescent cells to treat aging-related pathologies, in 2011, it was shown that eliminate specifically senescent cells in progeroid mice, so the white ones, contribute to improve their condition, and the same results were observed in naturally aged mice later. And to achieve this, they use a transgenic strain that can eliminate specifically cells that express anti-proliferative marker. And this analysis shows some benefits on various organs and function. It can decrease the risk of cancer and extend lifetime. So this observation raised several important questions. For example, if an healthy organism also already have this clearing senescent cells mechanism, maybe we can use it to eliminate more senescent cells or enhancing this immune mechanism in a therapeutical way. And also what's the place of senolysis in human therapy, because like cancer, the aim is to eliminate unwanted cells, but without affected healthy ones.
Today, there are promising targets of would-be senolytics, such as navitoclax and venetoclax. These two molecules are inhibitors of the BCL2 proteins that are located near mitochondria, which means that these molecules inhibit the apoptosis of senescent cells, but they are quite different. Actually, the venetoclax, it will inhibit directly the molecule of BCL2, but is not a potential senolytic in vitro. And navitoclax will inhibit three molecules: the BCL2, the BCLXL, and the BCLW. This is the strongest senolytic that is known, but it is limited in thrombocytopenia, and it is when there is a decrease in the number of platelets in blood. We're discussing how a senolytic might be developed clinically. The ideal condition would be in diseases that are highly asymptomatic, common, and where the use of senolytics would pose the least risk. Despite their high toxicity with these two molecules, we can make them less or have an acceptable toxicity by through new approaches like formulation or delivery or administration. Developing senolytics is quite challenging today because of the complications that may have a consequence. For example, rapid cell death that can lead to an impaired bonding or metabolic issues as seen in cancer treatment. Despite concerns, therapeutic trials of senolytics is possible, but there are different approaches that show us that senescent cells may not be the key for maintaining the essential tissue.
In conclusion, cellular senescence is a complex phenomenon that plays an essential role in aging and cancer prevention. Also, it can contribute to beneficial processes such as wound healing and tumor suppression, but its accumulation with age leads to detrimental effects, particularly through the secretion of SASP. This underscores the importance of continuing research on senescence to develop therapeutic strategies such as the senolytic therapy, aimed at eliminating senescent cells and reducing the negative impact on health.
Thank you very much. My presentation, so no doubt that the audience, you should have many questions. What are the questions you can ask your colleague, because they talk about a lot of different things, which were in the review, some that are not in the review. What do you think? Yes.
So you say there is a diminution of the risk of cancer, but there is also an increase of inflammation. So can it lead to cancer like in age? Then there is an accumulation, and then the inflammation can cause the cancer.
C'est quand même fou avec la présentation que tu viens de faire. Ok, in French.
Vous dites qu'il y a une diminution du risque des cancers, mais à la fois il y a une augmentation de l'inflammation. Du coup, moi j'avais vu des articles qui disaient que ça pouvait aussi mener à des cancers.
En fait, pour moi, c'est une question de quantité, d'accumulation. Quand il n'y en a pas beaucoup, en fait, finalement, elle va être bénéfique parce qu'elles vont avoir l'effet antiprolifératif qu'on recherche chez les cellules cancéreuses. Et quand il y en a moins, il y a une clairance immunitaire qui les élimine. Mais en fait, quand il y en a trop, elles vont juste s'accumuler et sécréter le SAS. Et ça, oui, par contre, ça va.
The question was about the fact that during cancer, we can have a forced accumulation of senescent cells. And if I well understood the question, it was about the fact that what is the relationship between senescence and cancer? And they are just replied that just answer that it's a question of balance. When the balance, of course, in terms of accumulation of senescent cells is too elevated, the risk is that you are going to have potential cancer through the SARS secretion, etc. So I don't know exactly if it's really the case because there is no proof concept that if you are accumulating, these cells are not cycling. So it should be an indirect effect of maybe of senescence to promote cancer cells proliferation, because if you start from the statement that senescent cells is not cycling, well, it's difficult to understand that these cells are going to be proteomogenic, you know. So I'm not sure that if they are really expressing P16, P53 or P21, if the cycle is really stopped, it's difficult to understand. But maybe indirectly by the secretion of pro-inflammatory cytokines, it should happen. But I never see that. Another question, comment? Yeah.
I just didn't understand what you said before on the 16th slide. What was the experiment done on a white mouse?
Which one, the 16th? Yes.
Yeah.
In English, right? Yeah, you're good in English.
By the technique of INCATAC, they eliminate only senescent cell in these mice, and the particularity of these white mice is they
White mouse. They are white on the screen, but don't think about
proteinuria her senescence is not natural. They induce this, and in order to observe the impact of this experiment, I think more rapidly. And after that, later, they observe the same results in the natural age mice.
Well, thank you. You're great. We are going to talk about that, okay? Next, with another presentation. You have to keep in mind, I don't know, I guess that in Master 1, maybe I told you that, maybe I presented the paper, you know, the first paper that came out from Vanderston, from the Minnesota, from the Mayo Clinic, that hypothesized that senescence, and particularly P16 positive cells, are the origin of all the complications associated with age. And the brilliant idea was to say that, okay, if we remove these cells, that means that the lifespan could be higher, etc., and no major issue associated with age. And in this paper, I guess it was published by in Science or Nature or something like that, a big paper, they really shown that finally, by removing these cells, and we're going to see after hope, I hope the next presentation will be a light hope. They really shown that without these cells, we are increasing lifespan, but it wasn't the main objective, but we are also increasing health span, okay? So that's quite important to see here in this slide. And I think that, Leah, you didn't say that at the origin. When you presented, if I remember, you presented the effect of clearing senescent cells and not inducing senescence. Or maybe I didn't understand, sorry, the discussion you had. But I think that this mouse line was at the origin of everything after regarding senolytics and etc. Okay, but we're going to come back on that. She spoiled a little bit the other publication. Next question. I think that we have a question here or comment. No?
No, it's for us.
Are you inspired by this kind of thing? I mean, that did you learn some stuff? What is there any dark side or
Did they found that other cells express P16 NK4A gene, or is it special of senescent cells?
What do you think about it? Do you think that the P16 expression is something that is 100% dedicated to senescent cells, or can we find some P16 cells that are not senescent? It's a question of definition. What is exactly senescence for you? When you consider that the P16 expression in cells is automatically associated with the senescent phenotype, or did you hear about some P16 positive cells that are not senescent? That's a good question. I don't have the answer. Maybe you have.
Well, in the article, they talk about one today. And they show that it was step by step. First, they found that there was an activation of the expression of P16, and that can help wound to heal. And later on, it's recently studied that the article showed that these cells that express the P16 are actually macrophages, which accumulate when we age, and when in the inflammatory phase of our healing.
Yes.
So I think, yes, I think P16 is also found in another type of cells.
So you mean,
That are not senescent.
That are not.
so you mean also that the origin of the P16 may be related to the macrophages, if I understand correctly?
Yes.
What is the question?
I mean, like, is the expression of P16 related with macrophages, like?
What do you mean? I mean, that it's related to the cell that you are checking. If you are checking cardiomyocyte, you can find some P16 expression in case of senescence. But here, what has been mentioned or maybe suggested by Fernanda is that in that case, precise case, I didn't know, P16 can be expressed without any other feature of senescence. So that means that to answer to the question, you can have some senescent cell, some P16 positive cell without senescence. But it's a little bit surprising for me. I try to think about. Yeah, you can have some big. Okay, let's come back to the beginning. Maybe P16, P21, P53. You know what it is? These are CDK inhibitors. So inhibitors of cyclin D kinase. The cyclines, you know, they are involved in the cell cycle, etc. The expression of this kind of marker or this kind of protein may witness the fact that you are stopping the cycle. When you stop the cycle, it doesn't mean that you are senescent. Some situations are borrowing also some cycling stop, but without any other feature of senescence. So I think that to answer to the question, it should be possible, but 99% of the time it is associated with senescence. But maybe in some situation you can find some P16 expression without any other expression of the marker of senescence.
I will add something. Actually, when they found out that, they thought that it's not the expression of P16 that directly helps the wound healing, but feature of these cells that are like senescent cells, because they also express, that helps this.
Yeah, but they are senescent. Finally, they are senescent. You just say that senescent cell. So wound healing, the connection between wound healing and senescence is something that is well known. For instance, you know, in my hand, we have at the lab we have the P16 luciferase maze. So that means that these mice, when you want to check P16 expression, you know what it is luciferase. You inject the luciferin, and you can follow the signal by bioimaging and biolight. Sorry. And finally, what is interesting in that guy, if you want to know if the genotype is a P16 luciferase or control, what you do, you perform a kind of smear, okay? And 1 day after, you come back, you inject the luciferin, and you will see that at the place you are performing the wound healing, you will see if it's purple or not. Purple means that under the gamma camera, of course. So that means that your mouse are, of course, borrowing the P16 luciferase construction. In case of not, that means that this mouse is wild type, okay? This mouse is wild type. So wound healing is associated with senescence, of course, because here again, it's you put that on the same slide as organogenesis, and I totally agree, because finally it's the same way. Organogenesis or wound healing, you need to initiate the process of cell proliferation and stop it. If you don't stop, that means that you will have maybe 6 fingers or 7 fingers or malformation, etc. So I told you earlier that senescence is crucial for the formation of the finger. So that's something that is well known. Here again, when the link, you have to stop the process and to stop it. If not, you are going to make a kind of chemogenesis. So that's why this kind of process, senescence is really well involved, and this is well known in this kind of process. Let's come back now to the question of today. Can we consider senescence? as a good marker of aging. I have a question, and after maybe you will have a question. Can we come back to the beginning and to the way how we can measure senescence? You proposed, Iman, I guess it was you, proposed different things, which were just before again before here. Okay, that's the characteristic of senescent cell. Can we just come back to be sure that everyone in the room are aware of what we are talking about? The first one is SAHF. Do you know what it is? No? Do you have an idea? You talk, Iman, about FOCI in English, I guess. Okay, you know what it is? What it is? DNA FOCI. It's like a kind of agglomeration of DNA. You know what it is? Accumulation of DNA. Because the DNA, the DNA folding is not really good in this sense. And that means that you can observe that by a microscope. So we did that in my lab a few years ago in microphages from human donors. And we observed that if you are just looking at the nucleus, of course, you need to label the DNA by, for instance, I don't know, any marker of the DNA, such as DAPI, whatever. And what's happened is that you can see this kind of point, you know, in the nucleus. So this punctiform stuff are very often associated with DNA aggregation, and this DNA aggregation is due to the fact that some proteins, such as histone, etc., are altered, and the consequence is finally that you will have a senescent phenotype. Not a consequence, but at least a connection between both. Okay. So first of all, increase of this FOCI, of course, is something that is very interesting because this is something you can measure, not easily, but this is something you can measure. Then you mentioned P16 expression. The next group will normally tell us that P16 is a protein, CDKN2A is a gene, okay, increased expression of the product of the CDKN2A locus. And in K4, we will see after why we use this. We can have also in this characteristic of senescent cell the increase normally of P21 and P53 or P19. There are other CDK inhibitor. DNA scarce. So DNA scarce is the mutation we talked about yesterday. Okay, about mitochondrial DNA, but this is something that can be also, of course, discussed with the genomic DNA. Do you know how we can measure this DNA mutation? Yes, you know. My question is not a question. Can you explain me how do you measure the DNA mutation? Not by sequencing, of course, because it's too expensive, too long, and blah, blah, blah. I would like you to propose something more easy to do. You've seen that in different publications since you are starting your master. What I'm talking about. You do that by histology. You can measure or evaluate the DNA mutation, double strand mutation. Okay, GAME-H2AX, you know what it is? This is a kind of strategy of antibody that is able to recognize the mutation on the DNA. Today, we are talking about considering senescence as a marker of aging. We need also the tools to evaluate this. I repeat, gamma-H2AX. You will see that in the publication, the use of this antibody in H2AX. It recognizes the mutation or the deletion in DNA. Okay, but there needs to be on the both DNA, on the double DNA. Okay, so for DNA scarce, of course, you have other techniques to try to measure that by using sequencing. We talked about that yesterday, but this is the more easy way to obtain a very good result. What about beta-gal activity? We talk very often about beta-gal activity. You know what it is? I hope. What is beta-gal activity? Why we have an increase of beta-gal activity?
Right.
Yeah.
Just go over this technique like briefly.
This is an antibody that is able to recognize, you know, the DNA double-strand DNA mutation.
Okay.
Okay. So you have that actually right now in your DNA, and normally your cells are going to go into apoptosis, etc. In that case, when the senescent cell accumulates, the DNA is can be altered, of course, and this is why the cell is going to turn senescent. And if you want to measure that, this DNA alteration, you can use the gamma H2AX technique. Okay, and this is done by microscopy technique. Of course, you need to have cells or you need to have some tissue. By histology, I think it's more difficult to assess, but this is something that is done. You can find it in the papers.
Okay.
You're welcome. What about beta-gal? Beta-gal is yeah.
It's called enzyme on lysosome.
Yeah.
And when the lysosomes are not destroyed but not really functional,
They are leaking. Yeah.
beta-galactosidase is on cytoplasm
Yeah.
and it's not normal, it's acidification of cell. And
Okay.
it's,
So you are using the fact, that you are measuring the fact that finally the cytoplasma is going to turn more acid. Why it turns more acid? Because with age, we know that the permeability of the lysosome is less efficient. And because as a consequence, we know that there is a kind of acidification of the cytoplasm. This beta-galactosidase is an enzyme that potentially works at a very low pH, not very low, but a low pH. So by this way, you can measure the fact that this enzyme is going to transform the X-gal, which is a substrate, into a product that will turn green. And by this way, you will see that if your cytoplasma is green, it's because finally it's more acid, and then it means that it should be associated with age and senescence. Okay, so the different steps are, first of all, lysosome leaking. If you have lysosome leaking and a decrease of the cytoplasmic pH, beta-galactosidase acts as with a very low pH. You add the substrate, which is X-gal, the name is X-gal, and then you turn into a green product that you can measure, of course, by microscopy. You should know that by heart, okay?
It is a green product.
At the end, yeah, it turns green. I don't know why exactly. This is a kind of blue-green. I don't know. Maybe I'm like blue-green. You don't have a picture of that? In my picture, they are green. That's and in my hand, they are not working. So because you have a kit which is provided by the cell signaling company, maybe you know that, very famous company. And it's not that it doesn't work, but you have some specific condition to get this kind of signal. I think that it works very well in cell line. As soon as you are using some slice of tissue, it doesn't work very well. So that means that you have to be in specific condition for assessing the beta-gal activity. Some people are measuring beta-gal activity through the use of flow cytometry. So this is a particular condition, but you can measure that. Yes?
To understand, so the beta-galactosidase is activated when the
pH is low. Yeah.
What?
When the pH of the cytoplasm is low. And that is low because the lysosome is leaking.
Okay.
Okay? And the lysosome is leaking because the cell is aged or is turning senescent.
Okay.
Okay? There are also other stuff that are not functioning very well in the aged cell. For instance, yesterday we talked about mitochondria. You should know that mitochondria has a kind of potential, on the membrane. We know that the potential may be modified by the senescence or by the aging of the cell. So, I mean that in the future you will be at the origin maybe of different discovery regarding this kind of regulation and alteration of the cell, and maybe one day we will have a very precise, because all these markers are not very precise. We will have a very precise and easy to do marker that will give us the information about. Okay, getting the information about senescence is something. Predicting the senescence, the future senescence is better. Okay. Then last, of course, it's cellular length. We talk about that on Monday, and decrease of proliferation. Other feature also of the senescent cell, you should know that. I think that last year we talked about that. The cells are large, bigger. You can have accumulation of different things. Lipofuscin, you know what it is? This is a kind of byproduct that should be degraded in in physiological situation, and that accumulates in senescent cell. So lipofuscin, etc., etc. You have different marker. no one is a gold standard marker, okay? Except it maybe the expression of P16 and the other ones, and BT-egal sometimes, but there is no gold standard to evaluate. You have to do different markers to be sure that your study is going to be or is senescence. Okay, I have another question about that. So SH, CDKN2A locus is done, senescence in with the way. Yeah, I think that Iman, you told us that senescence is always no. Can you come back to the first one? Because I want to be sure the other one. It was the first? No, so next here. Just to be sure that we are on the same page. As you can see, Iman told us that senescence can be induced by DNA damage, by telomere dysfunction, by oxidative stress, and by other cell damages. Do you agree with that? Yes, all of you? I think that we discussed already that senescence. Think about this process. The role of the senescence. What is the physiological role of senescence? To stop the cell cycle. Why?
To prevent the
Okay, to avoid the proliferation of, let's say just an altered cell. Okay. The alteration, the information that the cell is altered is done by who in the cell? Who is the first thing that is crucial to induce the senescence? What is for you the on-off button? How do you connect senescence with cell alteration? What is the main alteration that will lead to senescence? Yeah. You have to think about it. You have to think that you will find a lot of publication and in discover that the case also. This is not a mistake, but this is something that can be confusing for you. You will have senescence if you have DNA damage. That's a crucial sentence. You will have senescence if you have DNA damage. Okay. Then the question is now, how can we obtain DNA damage? First of all, could be telomere dysfunction. Let's come back to the workshop of last Monday. It should be oxidative stress. Let's come back to the workshop of yesterday with the ROS, etc. Okay. Other cell damages, maybe by UV exposure, whatever. Okay. But you have to retain that if you don't have DNA damage, there is no sense to have the senescence process. Okay, because the senescence process is a physiological process that happens when the DNA is damaged. Okay, so I just wanted to continue on this slide because it's important for you. Keep that in your mind. Then, blah blah blah. Oh, let's talk about the capacity of the reversibility or irreversibility of senescence. What do you think about that? Is it something that you can reverse or no? And what's happened, of course, if you reverse or if you are not able to reverse? What is your opinion? Maybe the group first and the audience next. Here you talk about irreversibility criteria. So that means that for you, senescence is something that is irreversible. Right?
En français.
Vous parlez ça veut dire que depuis le début, tu commences à tu dis que c'est irréversible. C'est un critère d'irréversibilité, la senescence pour toi. Est-ce que tu maintiens ça? Est-ce que tu peux nous en dire plus, etc. Juste, vous pouvez mettre le chat parce qu'il y a des questions aussi pour ne pas les louper. Voilà, à gauche, tu le vois là, placez-vous là. Il y a microscopie. Thank you, Yamina. And Mariam, again, to give the DNA temperature. Okay. My question is regarding the reversibility of the process. What do you think about it?
En fait, au début, quand il y a la sénescence, il y a des dommages cellulaires, etc., on va avoir l'activation des deux basiques, P16 et P53. Et en fait, à ce moment-là, entre guillemets, c'est la réponse initiale parce que en réalité, la cellule, elle a la capacité de pouvoir revenir dans le cycle cellulaire. Mais c'est le fait d'être exposé à plus de...
D'accord, il y a une notion de temps.
Oui, voilà.
D'accord, j'avais pas d'ailleurs. D'accord, ok. Donc, okay. So she just mentioned that this is a question of time, maybe. You know, on the first initial response, it's for them, it's possible that the cells, when they turn senescent at the beginning, on the first stimulus, they can reverse if the problem is fixed, maybe they can reverse. I'm not sure of that, but let's see, maybe in the review, I don't remember what they express here. But at the end, when the cell is submitted to a kind of long-term or middle-term exposure to different states, and of course an increase of cell cycle arrest, that means that finally the cell will turn senescent. I think that there is a discussion about the reversibility state of the senescence. Some people say that this is totally irreversible, okay? And some people argue that maybe in such different case, it can be reversed because of the cells, I don't know, DNA reparation or whatever, okay? I'm not sure that the time is really something that is crucial, but maybe, so let's just also amend. But according to me, you have to keep in mind that in our hand, and regarding the use of this senescence as a marker of aging, you can consider that the senescence is irreversible, okay? Because we are talking about a situation when you have prolonged exposure to different stresses, such as inflammation, such as growth, or whatever, and finally you will have that DNA damage, and the DNA damage, except in some rare case where you can repair, of course, you will have senescence and you will have long-term senescence, okay? So, for the definition, I will keep that it is something that is irreversible. Yeah.
Et aussi, c'est ça qui la différencie aussi notamment d'une cellule quiescente qui, elle, si on l'avait par exemple avec du USVF ou j'en sais rien, ben, elle peut retourner.
Yeah, by definition, the cell quiescence is something that is, in terms of time, this is something that is not so on the long time, you know what I mean? So this is something that is just temporal. Yeah.
Did they find the share-code of DNA damage to activate?
That's a good question. That's a good question. I think I will say yes. I already saw that, but in fish. I'm not sure that in human has been done. In fish model, we know that, and yesterday we talked about the DNA mutation of mitochondria. I'm pretty sure that if you reach a kind of threshold, of course, you will have some consequences, such as here DNA mutation. Senescence or maybe in the mitochondrial alteration, we talk about mitochondria, that will be critical for the cell itself, okay? So the notion of threshold, this is something that, according to me, exists in the field of senescence. This is not a kind of just progressive step. Okay? Other question on this slide? Yeah?
If there is a minor but beneficial mutation on the DNA, can senescence also negate it?
In terms of what? You mean that is beneficial, positive mutation?
Yes.
It's not mutation, it's deletion. Yeah. So that means that you have some DNA break, So here you are talking about mutation. Mutation, when you replace one thing by the other thing, I'm not sure that you will have senescence automatically. But I have to double check, but I'm not sure. If you have a gain of function, I'm not sure that your cell is going to turn senescent, because the cell is not going to have the information about the alteration of the DNA. Just mutation doesn't mean that it's altered. The alteration is through DNA double-strand break. That's crucial. Maybe when I explain you the use of gamma X, I miss that. You need to have breaks and not mutation. Okay. Reversibility. Who is in charge of cell clearance, of senescent cell clearance? You talk about finally aging is an accumulation of senescent cells. Who is in charge of cell clearance of these cells?
In this article, that clearance was made by month. Was it study?
By month? No, who is in charge? I mean that which cells are in charge of senescence? You mentioned that on the slide. Maybe go.
Macrophages?
Yeah. Okay. So immune system. Let's say that globally immune system. Okay. The immune. Yeah. Here. Yeah. Come back. On left. Okay. Okay. So if I understand what you are mentioning here, that means that at the end, that will mean that all we are talking about is just a consequence of the alteration of immune system. If immune system will do the good job, there is no senescence, right or no? What do you think about that? So can we talk about senescence or finally a loss of or alteration of immune system rather? That's a big debate. How do you think about that? So finally, it's quite easy to fight against senescence. We just have to educate the immune system, and that's it. Is it the case or no? Is it so easy? Maybe. What do you think? Est-ce que vous pensez que toute cette histoire de cellules immunes et que finalement, il suffirait de réaméliorer son système immunitaire pour qu'on n'ait pas de sénescence?
C'est là où il y a tout le débat parce que, par exemple, dans l'article, il est mentionné que oui, le système immunitaire, il va forcément diminuer avec l'âge, parce que ça va moins combattre les cellules sénescentes. En même temps, il y a une production, il y a un autre débat comme quoi ils disent que c'est la production de cellules sénescentes qui augmente énormément.
Ça, c'est vrai. Oui, non, on sait ça. We know that senescent cell production is progressively increasing. We know, because we know something, that we know that these cells are resistant to apoptosis. We don't know exactly why, but we know. What we know also is that in a physiological situation, these cells not could be eliminated through the help of human cells. So, of course, here again, this is a question of balance, okay? In one hand, you have a decreased capacity of your human cells to eliminate the cell, and on the other hand, you will have accumulation because they are more resistant to apoptosis. But the answer is yes. I mean that in my previous question, if you are able to potentialize your human system, that will be very interesting and in terms of senescent cell currents, because of course you will be able to at least eliminate with high frequency these cells, and that's quite important to eliminate these cells because you know that with age they will accumulate in your organism. Yes.
If the form the association between the number of senescent cells and the number of exhausted lymphocytes.
Do we know that? Do we know if est-ce qu'ils ont pu voir une accumulation de cellules sénescentes en corrélation avec, par exemple, une sorte de fatigue des cellules immunitaires, donc les lymphocytes et tout comme ça? So the question is, can we link the exhaustion of the immune system with the accumulation of senescent cell? Answer is yes. Again, why? Because there is a very simple thing to understand. The immune system is not, how can I say that, is associated with the organism. So that means that if the cells of the organism are going to turn senescent, the immune cells are going also to be senescent. And we talk about immunosenescence. The immunosenescence is the aging of the immune system. And by this way, you can correlate perfectly, not perfectly, but a loss of immune cell capacities with the occurrence of senescent cell, okay? Because we know that there is a strong relationship between both. So the capacity of eliminating these cells by the different lymphocytes or different immune cells. And that's why I told you that if you are able to, how can I say that, enhance your immune system as long as possible, that will be very interesting for you because we know that your immune system, if it is really efficient, it will fight against the occurrence of this. This is also the case really for cancer cells. You should know that. You know, the immunotherapy is based on this kind of properties. We can think about that also in the future for eliminating these cells. We can say that, okay, we're going to educate the immune system to be as efficient as possible and as long as possible to fight against this cell. Okay? To fight against the cells when they are, here again, pathological. Keep in mind that they present right now that senescence is not only pathology, not only associated with pathology. During embryogenesis, during different phases, it is something that is positive. You need senescence. At this time, here again, we have all senescent cells within our organism. Okay, but this is a kind of balanced ratio. Beneficial role, okay, sabbatical, okay, would oh. Excuse me.
I have a question.
Yeah, go ahead.
So basically, for instance, as we have described, it's mainly adult stem cell cycle. Is it really specific to a phase of a cell cycle, like at what phase does it really stop it? Yes.
I didn't listen to the question.
Yeah, I
You
know
have to answer?
sorry.
À quelle phase du cycle il y a l'armée cellulaire?
Okay, on which part, you understood the question? So you have answer?
I think it's between G1 and M, and S.
Yeah.
Population.
After G1.
After G1.
Yeah, you talk about barrier again infection. I didn't understand. What in a sense is a barrier again infection? I didn't know.
Yes, actually, the senescent cells will recruit immune cells to evitate
Okay,
the spread of
Okay, understand. Yeah, so here again, the same principle done before, okay. Atherosclerosis, I think that atherosclerosis is just an example here. We don't have to keep that in our head because we can use those. What about the spreading of senescence? Seeing that, Léa, you talk about the spreading of senescence in the tissue, or can you explain that for us? Or maybe you?
Donc ça peut être par acide.
No, I like
Like we can see, it can be a paracrine effect. Like, for example, if an LC cell is next to an senescent cell, it can be contaminating. And it can be also endocrine via the blood, because when the senescent cell secretes the sap, so they go in the blood and can access to a lot of other tissues, and this can lead to chronic inflammation environment and everywhere, and this can lead also to remodeling of the certain cell and matrix, and this.
Okay, that's good. What are your opinion about that? Do you believe that? Do you believe that just by secreting some IL-6, TNF-alpha, or whatever, MMP, the senescence should be a kind of contamination? Do you buy this theory? Yes or no? It has been shown. It has been shown. This is scientific fact that you have also to have your own vision. And what do you think about that? Yourself are continuously exposed to pro-inflammatory cytokines, and that's not the reason for them to turn senescent. So, according to me, because you don't want to give your point of view, but according to me, we can. This is a review, and this scheme has been really largely diffused, and people say that okay, cell-senescent breeding, something that can be associated with SARS production and protein effect and whatever. This is true, but that cannot be the only reason. I think that there are some other reasons. I don't know exactly how and why, but I'm pretty sure that the senescent cell is not only expressing some inflammatory cytokine. We know that it can secrete some metalloprotease, but I don't know exactly how and why. Maybe DNA releasing also something like that. I'm pretty sure that the senescent cell is going to how can I say that, contaminate other cells by other ways. I don't have the answer, of course. I don't know exactly how, but I'm pretty sure that it cannot be just a kind of pro-inflammatory signal. In English, maybe.
Like there is areas that more sensible, like in the adipose tissues or liver.
Yeah.
And maybe this connection with the SAS factor can
Tu peux encore insister.
du coup ça c'est
Oui, des fois.
l'accès de la goutte d'eau quoi.
Tu fais des trucs difficiles. Okay, so she just mentioned that depending on the tissue, of course, and this is, I think, a good idea, we can have a kind of threshold, and when the threshold is, of course, over, we can have the spreading of this. I think that this is not a so bad idea, the first part of the answer, because depending on the tissue, we just mentioned that the immune cell may have a crucial role in the senescence, not spreading, but senescence clearance of the clearance of senescence cells. Sorry. I think that in different tissues, such as liver, adipose tissue, the brain, etc., you know, the immune cells are not the same. They can be like more affected by aging, etc., etc. So that means that maybe some tissues that could explain also why. Maybe you don't know that. Why? What is the first tissue to turn senescent? If we can make a kind of chronological appearance of senescence. Do you know that? Have you heard about that?
The skin.
Sorry?
The skin.
The skin? I don't know. The others?
Embryonic tissue.
Embryonic, yeah, pathologically cell. Yeah, of course, embryon is a lot of senescence when you scale. Okay, but I mean that regarding aging process. What do you think? The skin?
The vessels.
The vessels, yeah. Another one. Which kind of tissues we are talking about? The other one is also the, I think I told you that, no? Gastric endothelium. Why? What is the particularity of these tissues? No?
It's a region.
Yeah, it's a high proliferative and high regenerative tissue. And of course, think about also, we didn't talk about that today because I think that for me it's admitted by you and because you follow my some course I gave you last year. You remember the clock of the cell clock? Remember that after a few regeneration, the cells are obligatory turn senescent? Remember that. So these tissues that are highly proliferative, you know, that's why these tissues are targeted during chemotherapy, for instance, when you have cancer. You have the epithelium of the gastric intestinal gastric, the skin, of course, all the hairs. That means that finally this high proliferative tissue can be exhausted before the other one and will turn senescent before the other one. Okay? So that's quite important to understand also that in this tissue, maybe the immune system is less efficient, will be exhausted before other tissues, and at the end, you will have more senescence in these tissues. Okay? This is in condition and basal condition. As soon as you stimulate the condition, for instance, you take some mice, this is what we've done in the past, you put them under alpha diet. You know what it is, alpha diet? You try to induce obesity. After 2 weeks of alpha diet, you kill them, you collect the adipose tissue. The adipose tissue is senescent. 2 weeks of alpha diet. So that means that if the tissue is very solicited here by alpha diet, remember what we talked about yesterday, mitochondria, metabolism, growth production, etc., DNA damage. Here again. When a tissue is highly exposed, or when a tissue is normally with high proliferative level, these are maybe caused to get, of course, high senescence in the tissue. Last thing, I don't know who said that, but someone said that it's difficult to measure senescence in human. I think that maybe when it was you, but the other also can. Why it's so difficult to measure senescence in human? Because today we need to state about the fact that we are able to measure senescence in human in the future. You have $1 million, you want to build a kind of clinic with the capacity to measure that. So what do you propose? Help.
Si on est à 1 million d'euros, j'essaierai de...
$1 million, not €1 million.
Mais comme on n'a pas vraiment de marqueur vraiment spécifique, ben, je m'attarderai sur tous les marqueurs.
Okay, so you don't have precise idea. Okay.
Parce ils voudraient, they want to target the SAS, but in function of what the consequences of the inter of the cells in senescence and in function of the tissue where the senescent cells is located, the factor of the SAS is not the same.
Yeah.
And so it's
So that means that you cannot just collect some blood and make some tests. Okay, so that means that you would love to get a kind of signature in maybe in blood cells or in blood, at least in plasma, that is specific from aging of senescent occurrence in one tissues. That's a good idea. That's some stuff that's ongoing, actually. Yesterday or on Monday, I told you that we are trying to identify the telomere length and the occurrence of senescence in leukocytes or in blood cells. And we are trying to connect that with aging on one tissue. For instance, here I'm working on skeletal muscle. But this is some ideas that are actually investigated. And this is not so stupid to think that in the future we'll be able to get some biomarker, of course, in the plasma ideally, that will be able to predict or to diagnose the senescence occurrence in different tissues. So, okay. If you have to measure right now the senescence in one patient, what do you propose? You in the audience, what do you think? What is the best marker for you? What is P16 expression? This is something that could be interesting. Of course, you need some biological material, such as maybe bulk biopsy, because this is done not on the plasma.
Use maybe the sample beta galactosidas or DNA.
DNA break. Yeah, but that means that you need a biopsy. Nowadays, there is no circulating biomarker of senescence. We don't know exactly how to measure senescence in circulation. Okay, so that means that we have to do that in biopsy or at least in the blood cells, in the circulating cells, but we are not able to do that. Some people argue that we can find some stuff in the blood that could be highly connected to senescence. You know that when the cell turns senescent, and then after finally it will be it will die by apoptosis, the cell is going to release something, and particularly some DNA. So maybe we can think about the fact that in the plasma, you have DNA in the plasma, circulating DNA. You can collect the DNA, make a kind of PCR in the DNA with the DNA, and try to see if you can detect maybe some motifs that are specific of cell senescence. These are also some stuff that are developed in Germany with the objective to maybe get some biomarker of senescence. Yeah. This is what we are doing right now.
We can.
Yeah, of course, and it works.
Okay.
It works. On blood cells, you can measure the P16 expression.
Because you told that if a bone biopsy is needed.
No, we are talking about some tissues, or if you want to see in the skin or whatever, but in the blood. But the blood is not going to inform you about which organ is going to turn senescent, if it's one organ or if it's the total organism. Okay, so that's the difficulty. Last thing that we are going to discuss right now, and this is something directly connected with the next presentation, is about the use of senolytics. I think that Fernando, you present something about that. Just one question. You know what it is, senolytics? You understood exactly? It was something before between 2015, 2017, maybe. I don't know. Do you have the date of the first clinical trial for the use of senolytics? Remember or not? I think it was in Newcastle, done by Von Zelinski or something like that, one of the authors. And to my knowledge, today there is no ongoing, but maybe a mistake, clinical trial on the use of senolytics. Why? It's because maybe the result can be a little bit lower than expected. And I think that Fernanda asked a good question. Cell death may lead to complication. You know, as I told you last year, the consequence of cell death by senolytic is highly risky because we understood that during scarring or during regeneration, you need also senescence. So it's difficult to make the difference between cycling cells that are physiological and senescent cells that are pathological. But in terms of proof of concept, that's an ideal tool because it works. I mean, that if you delete, if you remove the senescent cell, we will see in the future presentation, the next presentation, that it works very well. So that was a proof of concept.
Also, I have a question about white mice who they used senolytic. In your experience, you killed all the
senescent cells. And the mice didn't die if you kill all the senescent cells.
Because we are talking about something like, last year when you are in Master 1, I told you that you promote some kind of all in tissues, but it is not true. I mean, that you are targeting only 1% of the cells at the end of the day. Okay, but this 1% could be crucial also. So it depends on exactly what are the consequences. I'm not an expert on that, but I'm not an expert on that, but I'm pretty sure that that if you want to continue on the senolytic use, you need to be more and more specific. First of all, I don't know if you remember last year we did a work on how to try to build a senolytic, to consider senolytic, and we propose different cells to test, and we need to be specific on a senescent cell and not on other cells, etc., etc. Okay, so that means that you have to be really specific, and maybe ideally it should be to be specific of a cell type which is senescent. So that means that in the future, I'm pretty sure that you guys are going to work on detecting the senescent, but only in one cell type. For instance, I don't know, Farah is a specialist of skeletal muscle. Should we be in charge of detecting how we can target muscle cells that are senescent? And when muscle cells are senescent, they are harboring, of course, kind of marker, extracellular marker, that are not the same than the ones that are displayed by the adipose tissue. That should be something that, of course, this is not today something that we are working on, but in the future, I'm pretty sure that this is something that you might consider. Last one.
Can the use of the senolytic be associated with increased cancer? Because if I have to do the name correctly, that you are stopping senolysis in this case, senescence. So basically, we are stopping the cell cycle. So in other terms, we are increasing the proliferation. So this might be associated with cancer.
What do you think? Or what do you think? This is a good remark. Last year, normally, if you follow my courses, you have the answer. She mentioned that she said that there is something I don't understand. If you are treating your cell, that's a senescent cell with senolytic, that means that you are going to stop the senescence process. So that means that the cell is going to again cycle, etc., etc. So you are going to promote the tumorigenesis in cancer. Is that true or not?
Yes. No, because it's not the same cell. If there are senescent cells, it's not the healthy cell. So
she's not talking about healthy cell. She talk about senescent cell. Your senolytic is going to target the cell, stop the senescent process, and then the cell is going to be what again effective or not. But if the senescent cell is going to you watch. Yeah, I mean, the mistake is at the beginning.
I don't know, because you're targeting the senescent cell, so you are targeting beta, the beta galactosidase, the P16 senolytic cell. You are not targeting the other cell.
Yeah, but we are talking about the same cell. The senescent cell. She said that okay, if you are targeting the senescent cell by senolytic, that means that you have stopped senescence in this cell. Then the cells, the risk is to the cell to become tumorigenic. What is your opinion? What happened? Yeah, that's why. The senolytic is not going to stop the senescence process, it's going to kill the senescence. So that's why they are going to totally disappear. Not totally. Within charge of the next paper, they are going to show you that senescent cells are going to disappear. That's why it's quite interesting. But it's also very risky because, as we talk about, the risk is that these cells were maybe okay senescent, but maybe they were useful for the tissue. If you kill them, what's happened? And if you kill too much cells, for instance, if you kill too much lymphocytes in small time, what's happened is that these cells are going to release all their content in the bloodstream. So a lot of lipids and etc. You are going to have some side effect that will be worse than the senescent cell. So that's why it's quite important to consider also. And I think that during the last 10 years, people are thinking about that and try to fix the issue we can have with the use of senolytic. You should hear about xenomorphic and xenostatic. You know what it is? Come on. Yes, you know what it is. What is xenomorphic? What else xenomorphic? We talked about that last year.
Stopping the SAS.
Yeah, the xenomorphic is a drug or a medicine that will be able to fight against the consequences of senescence. Not killing the senescent cell, but only normally this review. This is not this scheme is not from the review, right?
No.
Yeah. In this review here, I don't know if you have the ref. You have one drug like that with senolytics, and the other one is with xenomorphic. So xenomorphic is a drug that is able to try to stop the consequences of senescence. And the last one is xenostatic. Did you hear about xenostatic? What it is? We talked about that last year.
So name it stopping.
No,
Static.
no, xenostatic, no.
Like,
We are in this is a kind of pharmacology consideration here. Xenostatic, no? A xenostatic is a drug that is able, here again, to try to fight against the senescence pathway, but by acting before the senescence. For instance, if you have what we call the mitoprotector, which is a drug that is able to enhance the mitochondrial function or whatever, that means that you will have less senescence because of yesterday's explanation. Okay. If you don't know, have some drugs that are able to increase the telomere length in one day, this is a xenostatic. You are trying to act not directly on senescence, not directly on cell life, but before the senescence itself. Okay, xenostatic. Thank you very much, Leah, Fernanda, and Iman again for the presentation. That's good in English. Next one. Who's next? Not you? Up to you. Which one do you have? The one that from the 6 maybe at the end and it's about right. I'm going to tell. So one from Darren Baker, I think first. Is that sure? This is not one. And we will finish. Oh no, maybe we can start by you. Sorry, I need 2 things. Yeah, in K4 and then are you? Sorry, I mean you all. I'm not the one online. All of them are.
Yeah.
Wait, there are also I need to check also the so okay. That's a good point, sir. That's a good point. Thank You try to find the mistake? That's it. Well, I'm not here. Yes. You want to make it on the desktop, or we can open the. Okay. At the end. Wow, nice background. So we have Fanta, we have Yamina, and we have Antoine. Only Kazim is here, and the other one are online? Not sure, Antoine is online? Mariam and Lona, your camera are off. So Yamina, you're going to present, Antoine, you're going to present, Pankaj, it's okay for you?
Yeah, it's good.
Yeah? Okay, so sorry again, because we cannot
Yes.
have our camera on. I have one people on the stuff, but she's not really know exactly what to do. So just propose me to do that on Zoom, but
En fait, c'est vraiment faire un webinaire.
avant, ça veut dire que ça avant de faire la caméra.
Non, mais c'est avant de mettre le coup.
Ouais. Sur Zoom. Et je sais pas si là, vous expliquez, non? Ce qu'ils ont fait, c'est des liens qu'ils ont fait. Si ça marche, peut-être. Ah ouais, mais comment je fais ça, moi? C'est vite. Ah, mais je peux pas, je vais pas le faire. Mon truc à moi. C'est un truc d'ici. Bon, laisse tomber.
Just Antoine or Panjak, who will be sharing his screen for the presentation?
Ananda
Because he's
Kazim
in
is sharing the screen
room.
there.
Yeah.
I'm using my iPad, so I don't have access to, I cannot share the screen.
Same as me. So whether it will be Antoine
problem I
Or
can
Kazim, yes.
Kazim.
Yeah, but I can if you want, but I think go for it.
You can share the G slide. I sent you yesterday.
Kazim, I think that Yamina is trying to say, no? You want to talk with Kazim? You cannot see him, sorry. But,
I just got slide from
I was just asking whether Kazim will share his screen.
You can also share if you want, because just 3 slides for me. You can also share your screen.
C'était pour ça. Sur les termes de la démarre, non?
Ok.
Attendez.
Et là,
On ne trouve pas, on ne trouve plus rien. Il n'y a plus. Yamina, elle est comme ça, c'est le bordel. Sur la quoi? Great. Okay. Now, and then I can play with the. Do you hear us? Yamina, do you hear us?
Yes.
Good. And now you can see us. Right?
Oh,
You see-
Very top view.
But if you want, yeah. Can, so you, yeah, oh. Okay, good. And of course, what is important is Kazim today. He's going to present. Okay, okay, so now. Do you see your screen online?
yes,
Yes. Allez.
yes.
Oui, c'est ce que là, il y avait du bruit.
Can I put the window?
Yeah.
Thank you.
Okay,
I want me to open here.
hello everyone, we are going to present the article.
Go ahead. Sorry for that. Here we go.
We are going to present the article name INT4A and IRF expression is a biomarker of aging now, and INT4A and IRF is proteins, and these proteins actually derive from the same gene, and but they have, just because they have different alternative splicing, they are responsible for different but similar functions. So INT4A is the actually acts as a cyclin-dependent kinase inhibitor, and they can inhibit the cyclin kinases, and by doing that they can halt the cell cycle, and if there is some kind of problem within the cells, and they can negate cells to go into the S phase, and so they can act as a tumor suppressors. And also IRF also function as a tumor suppressors, but they do it indirectly, and they can cause accumulation of p53 in the cells by interacting with another protein that degrades the p53 in the cells. So both of them only mostly repressed in the nucleus and becomes activated when the cells are damaged or in a cellular stress. And there is also trade-off. Actually, it comes to your question in the first slide. Even if they are tumor suppressors, they can avoid cells to turn into cancer cells within the tumor, within the tissues, but also they can cause senescence, and senescence cells can be dysfunctional and may cause inflammation. Also, they can also express MMPs that degrades the cellular matrix, and all of the things we have discussed in previous slides. And why did they choose these specific proteins specifically for their study? First of all, I must say that this article is published in 2004. So back then, probably they were using Drosophila and C. elegans, like lower organisms for their study to understand human diseases. And that's why they indicate that we should focus on higher organisms to understand the aging, because there is distinct difference between these lower organisms and higher organisms, because human tissues have stem cells. On the other hand, the other lower organisms don't have it. And stem cells are the most crucial cells in aging. And in previous studies, they have showed that these two proteins can regulate stem cell function, and if they increase in stem cells and other cells, they can reduce the function of stem cells. And so at the end, it causes aging. And they also showed in a lab that lots of cells, keratinocyte macrophages and fibroblasts collected from marine and human cells, they express high of these genes in terms of aging. So that's why they choose these gene proteins to look for if it's expressed in whole body. They want to see these markers should be expressed in whole body, in all tissues. And if it's expressed in all tissues, then we can say this is a good biomarker for aging. And also they want to evaluate the impact of caloric restriction. So they want to find a link between caloric restriction and this expression of these genes. And after that, they also interested in the regulatory factors that either increase the expression of these genes or reduce it. But their main goal is to find gene biomarkers that can be found in all of the tissues, so that we can, let's say, by just looking at one tissue, and this is an aged tissue. Yeah.
So to complete these objectives, they wanted first to characterize the expression of NK4A and ARF with age. So they did an RT-PCR, and they found out, as you can see in the figure A, that there is a big increase in about the ratio between old and young. There is a big increase of P16 NK4A and ARF when we compare to the other protein, which are not. And then they wanted to see if this expression increase was shared with all of the organs. And what they've seen is that this increase is the largest in kidney, ovary, uterus, and heart. And what we could say that it seems to be shared with all of the organs, as you can see with the clearer color, which are the yellow and the blue one. They are almost always higher than the darker one, which are the young rodents. Casimir, you can pass, please. Okay. Then they wanted to know if this increased expression was subject to the tissue-specific differences. So they did an immunohistochemistry on different parts of the body in the islet, uterus, and the spleen. And as you can see, the antibody that they used against the P16 protein stains more in the wild type old each time, but we can also say that it stained in the nucleus and in the cytoplasm. I'm sorry, they're working. And moreover, we can also see that this expression varies by tissue compartment. It's not clearly the same between, for example, the islet and the spleen. And then in the figure B, they analyzed the ratio between old and young, and they found out that there is a difference, and a tissue-specific difference, and a localization difference between the organs. For example, there are more expression of NK4A in the stroma of the spleen and in the stromal lymph node. Okay. Can you please pass? Then they wanted to know what could be the effect of the caloric restriction on the expression on NK4A and ARF. And as you can see in the figure A, it seems that the caloric restriction reduces they decrease the expression of NK4A ARF and significantly. And this will mean that it lowers the age-induced expression in the specific organs that they study here. But as you can see in the figure A, this observation is not shared with all of the organs because it's significant in the kidney, the ovary, the heart, and the testis, but it is not in the lung, the liver, or the lymph nodes. Finally, they wanted to establish the link between NK4A ARF expression and the senescence marker SA beta-galactosidase that we've seen earlier today. So they found out that the expression of, well, the activity of SA beta-galactosidase correlated with the NK4A ARF expression, which means that higher NK4A ARF levels in aged kidneys, which because we're looking at the kidneys right now, matched with the increased SA beta-galactosidase activity. And when we're looking in the different groups that they made, we can see that the caloric restriction reduced both the NK4A ARF expression and the SA beta-galactosidase activity, and particularly in the kidney cortex.
Kazem, can you pass, please? Okay. So actually, to uncover regulatory influences on NK4A ARF, they studied in this article ETS1, which is a key transcription factor regulating this process. So they found that ETS1 expression correlates specifically with P16 NK4A, but not with ARF, so suggesting that it may selectively regulate P16 NK4A in response to age-related stress. So this finding introduces a possible age-related regulatory pathway involving ETS1 in this process. Can you pass the other slide? Thank you. So additional regulatory factors in NK4 ARF expression in this article, they shown that research suggests that the co-expression of P16 NK4A and ARF points to other regulatory factors influencing their activities, which are unknown. So while ETS1 specifically regulates P16 NK4A, it does not impact ARF, so indicating that both proteins likely share additional yet not identified co-regulators. So the strong correlation, as you can see, implies that age-related stress might activate these unknown factors, impacting aging processes and senescence across various species, various tissues like it was studied in this article. Can you pass, please? Thank you. So further validating their findings, like similar NK4A ARF expression patterns were observed in both mice and rats, which suggests that NK4A ARF's role in aging is conserved across species or expressed the same way at the same level. So they've done this cross-species to highlight the relevance, strengthening the potential of NK4A ARF as a universal marker of aging or as a marker that can be reproducible in species like mice, rats, etc. Can you pass, please? Okay. So in this slide, we are highlighting their comparison about caloric restriction versus growth hormone receptor deficiency in aging. So interestingly, while the caloric restriction reduces NK4 ARF expression in the kidney, growth hormone receptor deficiency has minimal impact, implying that this caloric restriction and growth hormone receptor pathways may influence aging through distinct molecular mechanisms. So this comparison provides new insights into how different genetic and environmental factors modulate this aging process in this study.
And for the discussion, INK4ARF as an aging marker. This research shows that as we age, the level of INK4ARF increase in various tissues. This makes it an useful marker for an aging, helping us to understand biological age, which is different from the chronological age. Caloric restriction, CR. This study have shown that the CR caloric restriction can have the lower level of INK4ARF and the other marker of aging in several tissues, which suggests caloric restriction could be a way to help to reduce the changing later to aging. Regulatory factors. One of important factor is ETS1, which helps to control the expression of INK4ARF. However, there are likely other factors which we do not know about that could influence the level of both PINK16, K4A and ARF. Next slide. Moving on the impact of INK4ARF on tissue regeneration and aging. We see that high level of INK4ARF can limit the ability of stem cells to regenerate tissues. This restriction can lead to tissue aging, organ failure in older organisms, showing how important this marker is in the aging process. Now, let's discuss the caloric restriction effects on INK4ARF. It has been found that the caloric restriction reduces the expression of INK4ARF, which is linked to few age-related health issues. And this suggests that caloric restriction might slow down the aging by preventing the buildup of old and non-functional cells, helping to keep our tissues healthy as we age. Can you switch the slide, please? Regarding ETS1, it specifically influences the expression of P16 INK4AF, but not ARF as previously mentioned. And this indicates that the ETS has focused effect on certain aging pathways. Additionally, ETS may be activated by stress signal related to aging and pointing its role how our body responds to stress as we get older. We have a hypothesis about unknown co-regulators. They are likely additional factors that control both P16 INK4AI and ARF expression separately from ETS1, indicating a more complex network regulation. And future studies should aim to identify these factors to better understand how INK4ARF levels are controlled during the aging. When we compare this to other aging model like growth hormone receptor GHR deficiency, we notice some differences. GHR deficiency does not significantly change INK4ARF level in most tissues, suggesting that caloric restriction and GHR pathways may influence aging in different ways. Now let's talk about the clinical uses of INK4ARF as an aging marker. There are several potential applications, such as using INK4ARF level to assess physiological age in clinical setting. This could give us a better understanding of a person's health beyond just chronological age. Additionally, measuring INK4ARF could help predict how well patients can regenerate tissues, which can guide treatment decisions for older patients. INK4ARF monitoring level could also help evaluate the effectiveness of the anti-aging therapies. Antoine?
Yeah, so to conclude, we share with you some key takes, some key points to remind you. So INK4ARF expression could be a robust biomarker and a possible factor of aging. The caloric reduction of INK4ARF highlights its potential to reserve tissue function, and it supports the use of INK4ARF in clinical age assessment and anti-aging intervention.
Thank you. Just, not maybe stop the share because I want to see one thing. Okay, what are your question, remark, commentary, etc.? What do you think about this publication? Do you have any question, any precision to get? Maybe because it's difficult to understand sometimes. Yep.
Can you explain me if there is a link between P16 and ARF or not?
What is the link, Kazim, between P16 and IR? Why is it they connect?
They are actually generated from the same locus on the chromosome, and they are also a shared common gene, but based on the promoter and alternative splicing. So we see two different proteins, but they have same function almost. In both of them, it's more repressor genes, but their the pathway they affect is different. The way they work is different.
If I can add, actually, the INK4-ARF locus encodes two tumor suppressors molecules, P16 and ARF, which are principal mediators of cellular senescence. That's why it was studied in this article.
Okay. There is also another.
I'm changing something for you. Really?
I don't know.
I'm just changing the camera. I didn't know that.
They also tried other sequence-dependent inhibitors for this study. They are all from the same family.
Other question, of course, was a commentary, yeah?
Why do they look like the growth hormone receptor specifically and not another thing?
What is the relationship, finally, between growth hormone receptor, or growth hormone itself, and aging or senescence? What can you say about that? Why did they choose to assess their modeling in this field? What did I do?
Guys, you heard the question.
Maybe not Kazim. The other one, what do you think, Antoine? I don't know,
I'm sorry, I didn't catch the question because I'm
What is
having some
why
issue.
is it choose to work with GR's growth hormone receptor? What is the relationship between growth hormone and senescence, finally? What was their hypothesis? That's a good question, but I think that you guys should have it, even in the audience. What could be the relationship between, even if you don't know, if you didn't read the publication, what could be the relationship between GH production or action and senescence?
Because the receptor production decrease with age.
Yes, but yeah.
I don't know.
If I can add, like GH
Yeah,
increase oxidative damage and low GHR reduce it, which limits cellular aging. It's like a very complex pathway. That's why they choose GHR, at least in this publication context. And they talk about like the external stresses, and one of them like could be oxidative and some other, and they choose GHR because GH can be how would I say, increased during the oxidative damage. That's I'm sure of.
I agree, Pankaj, but I think this is a consequence. If they use this model, yeah.
Maybe because GH increase the cell division, whereas P16 stop cell division.
They are trying to use a kind of model that will be able to promote, of course, cell proliferation, cell division, etc. So the growth hormone is the main role is to promote the cell propensity and the cell proliferation. So if you use growth hormone receptor deficiency models, that means that you want to decrease this effect of, or to at least to blunt the effect of GH, and to see what are the consequences on senescence. That's finally a complementary model of caloric restriction. Yesterday, we talked about the fact that mitochondria should be unloaded by strategies such as caloric restriction or whatever. You can also think that by acting on the one axis, which is IGF-1 insulin growth hormone, of course, you are going also to decrease the capacity of the cells to cycle, to divide, and finally to use some substrate. So that's why Pankaj, I think that In terms of oxidative stress, this is a consequence of the absence of the receptor of GH. And that in the main hypothesis, the GH deficiency, the GH pathway deficiency, is there, according to me, to try to mimic a kind of decrease of tropic signals. Okay, you understood that? That's important. They use two different models. I have a question, maybe, since if you can come back, something like, you know, on the histology when they, not this one, this one. Well, we cannot zoom in, but what do you think about the distribution of P16 here? So, do you know how we can analyze this picture here on the upper side? You know, what are we checking here? What is positive signal on this picture?
Expression of the.
Yeah, where is the expression? You can see. This is the purple one that you are doing. Wait, here, really? What are we measuring? The purple one is what? No. Where is the expression? What is the color of the expression of P16 here? Yes.
The black spot.
Yeah, it's not the black, but the brown one. This is peroxidase activity. And you should know that this is hematoxylin eosin staining in purple and in blue, of course. And then, so you can, of course, label the nucleus and membrana, etc. But you want to see P16 expression. So what to do? You use peroxidase experiment where the antibody is coupled with the peroxidase enzyme. But it was not my initial question. So now you know that. What is your opinion about the localization of P16? Where it should be, where it is? What is the reason? You guys that work on the paper, what do you think about that? They discussed that in the paper, but I don't remember exactly what they say about the localization.
Well, they say that the localization was both in the cytoplasm and the nucleus.
Yeah. This is something that was expected by you?
Not really.
Where we should find, where we are expecting to find the P16 protein? In the nucleus or in the cytoplasma?
In cytoplasma.
Really?
I don't know.
We are talking about inhibitor of CDK. Okay, so when they are active, they should migrate, of course, into the nucleus. Here they talk about a common repartition, as mentioned by Antoine, I guess, on cytoplasma and the nucleus. Sometimes the nucleus, sometimes the cytoplasma, depending on the tissue. So we can see that finally, maybe also this is something that we can note, that the localization of P16 expression should be also a kind of biomarker of aging. If we were able to discriminate finally the activation of P16, because P16 is there, but the question is P16 in the nucleus is different than P16 in the cytoplasma, and the impact and the role of this protein should be different. If it's in the cytoplasma, this is just a protein that is here, maybe. I say maybe because I know that is not the case, but waiting for an activation and migration into the nucleus to block the cell cycle. You have to imagine also that P16 has a role as a CDK inhibitor, but all the roles of P16 have been mentioned in the literature. You should find it. I don't remember exactly when, and which role, sorry, but you have to understand that this is not the only role. Of course, we are working on this role of CDK inhibitor, but also role have been pointed for the protein. Can you explain what is the figure B here? Because we see different bones in the bone marrow and spleen in the lymph node, and I think that you went a little bit fast on this figure. What did you understand in the audience here? Do you understand what they want to mean? What is the figure B? What is the message of the figure B? This is something we already discussed a few minutes ago when we talk about the review. What it is? All young, this is a ratio, bone marrow, okay, spleen, lymph node, moelle osseuse, heart, lymphatic, and then the total, and then the lymph positive, lymph negative, stroma, cells, B22, 0, blah, blah, blah. What it is? What do you want to know? tissue expression, yeah, but
Whether P16 expression is tissue specific or no.
is it only tissue specific? What do you mean about tissue specific? Why, for instance, for the first one, bone marrow, why is it try to study the expression of P16 in lin negative and lin positive? What it is lin negative and lin positive?
Cells.
It's different cells finally within the tissue. So they are not only saying that P16 is expressed in bone marrow twice or maybe 3 times or 4 times more than in age than in young. They also try to explore the discrepancy we can find between the cells that are composing the tissue. As you can see in the spleen, that's quite interesting because in the spleen, the stroma cells are positive, highly positive, compared to the cells that are not in the stroma, I guess, or even other MAC1 positive cells. So that means that it doesn't, it's not because your tissue is senescent that all the cells in the tissue are senescent, okay? So that's quite important also to notice that, and by this publication, it was the first time that we can see that, of course, in the tissue, all the cells are not going to turn senescent in the same time. So this is something that is maybe also, okay, after that, can discuss the fact that, as mentioned by Leah, you know, the spreading of senescence. We don't know exactly if the spreading of senescence, as we can call that, is a spreading which is tissue specific. You know, some cells should be maybe highly sensitive to senescence signal, or that could be directed on different cells, etc. Okay, on all the cells of the tissue. As I mentioned to you before, endothelial cells are going to turn senescent very rapidly. Okay, one of the most important cells that are senescent in organism. Other question? Other remark? I have a different. Did you discuss about the model? They use young and aged mice, I guess, or rats. I don't remember. It was mice or rat. I would like you to discuss about. Yeah, Antoine, go ahead.
Yeah, they use it depends on the experience.
Yeah, rodents.
Yeah, they use rodents.
What about the age of these animals, and what about the gender? Do you remember the age? It was 2.5 months for the young, 26 months old for the age. Is it something that for you is comparable or no? And you in the audience. What do you think about that? 2.5 months. If we are regarding, I don't remember if that was rat or mice, they use maybe both. For mice, you know, that if you want to compare young and age, of course you can use 3 months old versus 24. But it's quite different. It's like you are comparing two different space families. Okay, so it's quite different. It's quite difficult to compare this kind of mice.
Yes, another article that we saw, they use young mice, old mice from 18 to 20.
Yeah. It depends on what you want to see, but I'm just discussing the fact that using very young mice, such as 2.5 months, is very young, not very young, but young, is difficult to conclude. I think that in the future of studies, you should also have kind of some middle-aged mice or rats or whatever. That could be very informative for you. And what about the gender? Is it only male or they are using female? I don't remember.
I think it was equal number of female and males. And also they made about since they used very young mice, they couldn't detect expression of these genes in some tissues. That's why it's not good for comparison or test.
Yeah, that's why it's difficult sometimes to. That's why it's better to see if at 8 months old or something like that, you should find also. I think this is something important. And you can see here that what we discussed before, that different tissue are expressing senescence at different level and different time, etc. Stroma cells, so we discussed about that. So it was already by Antoine. Yeah, just a word about the EPS1. Can you just come back on EPS1 and why is they raise this target? What is the rationale? What is the scientific rationale for that?
So ETS1 is specifically associated with the regulation of P16 INK4A. and it's
Yeah, but
activated.
my question was how they identify this target? What was the scientific rationale?
How they identify it?
Yeah, because you are just saying that ETS1 is correct. Is okay. I understand. This is a kind of a consequence of the publication, or they just said that we know that it has been previously observed that ETS1 correlates with P16. I don't remember if they are using other source of information.
In the introduction, they mentioned about this, and that I think it just because others have found in other organisms, this transcription factor increases expression of this gene. That's why they decided to check it.
Okay. So it was something that they already identified. Okay.
Yes.
Another question on that, maybe to continue the discussion we had in C and in the figure C. Yeah, I did not remember we did that, but what it is exactly? Can you explain slowly for us what it is? What did they do? What are these tissues and
liver tissue?
What it is?
The liver tissue, right?
Liver?
No,
no, that kidney, no, maybe.
I don't know.
You remember who present that? I remember.
This is
Yamina?
Yes.
You presented that, but I think it was a little bit too fast on this part because we didn't have the time to follow you. Can you just redo the figure C and to have the question that was raised and the result we can observe because we don't know exactly what it is.
So actually, it was about the kidney. They marked P16 and the co-expression of P16 and ETS1.
What we are checking here is in blue. It's the expression of P16 in C in the figure C.
No, they're checking the essay beta-galactosidase.
It's a beta-gal.
Yeah.
Okay. Beta-gal.
Yes.
And what about the just on the bottom, the picture which is on the bottom? What it is? This is adipose tissue or what in rat? These are maybe piece of kidney. That's it.
These are kidney tissues. Part of
Kidney tissue also. So again, I'm the relative expression ratio. Blah blah blah. This is the G and the C. This is renal cortex. Okay, I didn't remember. Beta-gal staining in aldibiton and ferroextrusion mouse and rat kidneys. Okay, C
Yes.
renal cortex and renal medulla, and syn tissue slide was staining for mice. Okay, so you can see that we are both agree because for mouse this is blue for the beta-gal staining, and for rats it's on green, right or no? Or you see that blue
Yes.
every time?
Yes.
Okay, so that means that sometimes it's blue, sometimes it's green. Okay. EPS1 is done. The conserve across species, I'm not totally agree because even if they mention that when you compare mouse and rat, you are not really, of course, you are changing the species, but it's not totally. If you have this kind of regulation in human or in fish, between vertebrate and invertebrate and etc. Okay, but here, mouse and rat.
Yeah, actually, so it was stated in the article that they can conclude that this biomarker is universal because they compared rat and mice. But actually, I find that this is a limitation of the article because they didn't compare in other species or in human. I think which is a perspective to do after.
Yeah, I can understand. And that's totally right that you mentioned that if you want to make a biomarker as universal as possible, you have to validate it in different spaces. That would be great if you can do that. That means that the process is conserved across the spaces, and it is a strong process and a strong marker, okay? Another question, another remark, a final comment. Is it okay for you? So here we just remark that P16 expression is driven by INK4 gene that can be associated also with other genes that are on the same locus, such as ARF. And in this situation, we can follow up, of course, the expression of this protein in different tissue, in different conditions, etc. And you can revert by character restriction. So this is something that is totally agreement with what we mentioned yesterday. Okay. And then last but not least, And thank you very much again for Kazim, because he was alone. It was difficult to answer to the question. Last but not least, we are going to see what is the consequence of the absence of these P16 cells in the different tissue and the construction of mice. Okay? Okay, thank you very much. Like, I think you are great. Everyone along, all your friends are, you know, it's easy to be under the just the camera. Yeah. So Emmanuel, Salim, Samuela, and Hugo are going to present the last one. Are you going to go there? Okay. Who start? Emmanuel? You start?
I'm sorry.
You start in French? No, okay.
Okay, so I will introduction.
Did you share? Did you share with the other one? Maybe you can escape. Yeah, and then go to the Zoom one. Yeah, and
Oh my gosh.
on the road right if you want. Yeah, great. Here we go. The last one.
Oh, okay. Hi, everyone. Good afternoon. So today my team and I will be presenting on naturally occurring P16 INK4A positive cells and how they shorten have healthy lifestyle. So before we get into the results of this paper, we first have to go into what is cellular senescence. You guys have seen it from the other presentations, but in a nutshell, cellular senescence is a hallmark of aging that results in the stopping of cell division as a result of various cellular stressors, such as oxidative stress, DNA damage, the accumulation of it, introduction of genotoxic drugs or oncogenic drugs, as well as oncogenic stress. And as a result of those stressors, you can then be telomere attrition in the cell, as well as cyclic arrest, SAS, for the significant phenotype, as well as production of tumorigenesis. And one major point to remember from all of this is that the accumulation of senescent cells over time can negatively impact both lifespan and health span. Okay, so now to introduce P16 INK4A, you guys also just saw that, but essentially, this is one or two tumor suppressor proteins encoded by the CDKN2A gene. What this protein does is essentially distinctly stops the activation of cyclin-dependent kinases as it is a cyclin-dependent kinase inhibitor. And what this does downstream is result in the cell cycle arrest that is characteristic of senescence. Okay, so what was the goal of this paper? The first goal of this paper was to ensure that, or just to identify what is the role of P16 INK4A positive cells in aging, as previously the authors did the experiment in procreated mice, and they were able to see that clearance of these cells improved age-related decline. So then they hypothesized that therapeutic removal of these naturally occurring P16 INK4A positive cells could be beneficial in extending both lifespan and health span. To investigate this, there were three major aims. The first was validation of the mouse model that they used, as well as looking at the ability of a special drug to have that model act in regards to how it would clear out the senescent cells from inguinal white adipose tissue. Secondly, they wanted to make sure that, okay, once these cells are gone, as much as they could be, are there any adverse effects in regards to physiology or the function of these mice? And last but not least, they wanted to bring it back and just further investigate how these senescent cells can impact aging. And as I mentioned before, they used mice, specifically wild type C57BL6 mice, expressing the ATAC transgene, and they were various ages. And I guess a bit of a preview, but you guys will see our figures soon. So just for simplicity, divided these results up into what they found concerning these P16 INK4A positive cells, and then what happened after clearance. So these cells are found to accumulate over time with age, as mice were analyzed from 1 year, 12 months, up to 16 months, or into death. Also, there was promotion of tumorigenesis, as well as functionally impaired vital organs, namely the kidney and heart. There's also decreased
cardiac stress tolerance. Is that in the future cardiac aging as well as increased cardiac mass, mainly in the heart, as well as shortened lifespan and acceleration of death. On the contrary, when they were finally able to clear out these cells, there was increase in median lifespan, which ended with sex. There was delayed tumorigenesis as well as preserved organ functionality. And last but not least, the thing that was interesting was the benefit of cutaneous wound repair as a result of AP treatment.
Okay, so let's begin with the first figure. So as Emma said, the goal was to validate the model. So here they first performed a FACS to determine if there were GFP plus intensity on our attack mouse model, which is the case here. On the panel B, you can see the expression of senescence associated related genes on GFP plus cells, confirming that the attack model induced descending of the cell. And finally, here you have also a significant increase of the SA beta gal proportion of cells. No, a significant increase of SA beta gal positive cells in the GFP. So then they try to focus a little bit on the adipocytes, and here what we can see on the LD is that you have GFP plus cells proportion that is highly increased in the fat progenitor cells. So this basically means that you have a higher level of senescent cells, while in the non-treated attack condition, but once you treat them with the AP treatment, you can see a significant decrease, meaning that the clearance occurs in this particular case. Then you have two different pictures showing the attack, showing two inguine white adipose tissue and epidemial white adipose tissue at 12 months to 18 months. So without treatment, you can see that there is an increase of the SA beta gal phenotype here, for example, because you can see that the color is getting greener. And once you treat those with the AP treatment, you can see a decrease of this color, suggesting that the clearance is eliminating all those senescent cells. And so here they looked at the XGL crystals, so I don't have to remind you what this is, except that here, in fact, they are blue. And so you can see a significant increase of these XGL crystals in non-treated condition at 18 months, which translates to the fact that there are an increase of senescence in these perivascular cells, just here. And of course, again, the AP treatment is significantly decreasing this presence of XGL crystals. And as you may know, so when you are aging, when an individual is aging, there is this loss of fat mass, also the adipocyte atrophy, and the decrease of adipogenesis, and it is clearly shown here with first the decrease of fat mass, adipose diploid weight, and mean adipocyte diameter in the non-treated condition, and when you treat the mice with the AP treatment, you have the correction of this significant decrease, showing that the clearance of the senescent cells are preventing the age-related loss of at first the fat mass and secondly the atrophy. And here you have so two transcriptional factors, PPARG and CPPA, that are factors promoting adipogenesis. And as you can see in the white adipose tissue, you have a significant increase of these two transcriptional factor, which means that you have a decrease of adipogenesis, as we talked before. And this is also corrected with the AP treatment, and I did not precise that this was performed independently of the sex. So here you have the male in blue and the female in black.
Just before you switch, can we interrupt?
Yes, of course.
Because it's a long and very interesting paper, and I think that we need maybe some precision. Do you have any question on the first? This is the first, only the first figure, okay? This is an actual paper, so this is a big paper. You should have some question, I guess. Even if it has been well introduced, I think that if you didn't read the paper, you should have some question. Yes?
What is the AP injection you mentioned?
So yes, so the injection.
Why is they inject this AP drug? What it is?
The AP20187 injection is basically a homodimerizer. And it basically comes from the FKBP domain, and it like initiates protein-protein reaction.
Do you remember, maybe again last year, I presented the first paper. This is the second one from the Vanderson team. And I told you that they have the originality to induce the death of these cells, P16 positive cells, by expressing under the control of the P16 promoter, a caspase, the caspase 8. But if you do that, that means that we just talked about that at the beginning of the afternoon. You are going also to disrupt all the processes that are depending on senescence, such as organogenesis, etc. So that means that you have to keep your system inducible, and by this way, by using AP, they are going to use to give the possibility to induce when you want the expression of caspase 8 only in P16 positive cells. Okay, but it's quite important, of course, to understand why they are using this drug AP, which is a okay. What about the other things? So now you have, maybe you can just come back and go on the previous one. Yes, and the one, yeah. Okay. Can you just re-explain, Hugo? It was you or it was Manuel? I don't remember. Yeah. The GFP stuff.
Yes.
Why is there doing GFP? What is GFP positive?
So GFP positive cells mean that the P16 gene is, from what I understood, is correctly induced in the attack mice.
Mm-hmm.
And so here, from what I understood, it's like in the wild type and the attack mice, you can see the GFP negative intensity that is quite high. Not all of the cells are senescent. And here you have the GFP plus cells that have a bit. So I don't really know if it's significant or not. And so this proportion of GFP plus cells indicates that there is disproportion of senescent cells in the attack mouse, right?
Okay, I agree. But how do you switch from A to B now? How do you explain that there is a GFP negative and GFP positive? These are the cells that are positive. Finally, the remaining cells that are on the GFP positive here, please, are collect, and we can measure the expression of P16, caspase 8, etc. on these cells.
Yes.
And that, what does that mean? What is I don't remember if they discussed that, but
since it's a comparison between the GFP minus and plus, you have here a significant increase of all these senescence related.
Is this something that we are expecting? That?
I think so.
Yeah, you're right. Yeah.
But in my opinion, maybe they should have put these two before this one. I don't know, like to.
Yeah, I agree. But I'm not going to criticize this team. You know, this team is fantastic. So
yeah,
they are terrific. What about the positive cells? Some cells are still positive.
Yes.
What does that mean? That means that the system doesn't work at 100%. You are not able to kill all the P16 positive cells, right? So
Yes, for sure.
Yeah, you have cells. You can see here that you have some cells that are P16 positive in green. You can see because they are GFP positive. Finally, they have been finished like that. Finally, you know what I mean? Okay, they have been collect like that because they are GFP positive. And you can see that these cells, and this is a very consistent with, they are P16 positive. GFP is coupled with P16 expression, but they're also caspase-8 positive. What does that mean? They are not dead yet, but they are dying finally. These cells are going to die if you, I don't know, if you wait maybe some hours or some days, they're going to die. So that means that the system works very well, okay? Even if we don't have 100% of cells that are killed, and this is something that you can understand, cells are renewing, and finally some cells are going to escape to the system. And the figure C is confirming what we are talking about, okay? These cells are SIR positive, so this is something that we discussed. And here the signal is green, right? Not blue, green. Yes. In comma that is green. Okay, and then what about the next figure? They use adipose tissue. Do you know why? These guys are not expert in adipose tissue at all. I don't know if James signed the paper. James Kirkland, I think yes. No. Okay. So there is no reason at all for them to use this tissue. Why they are using this tissue?
I think I saw it in the paper.
No, they don't explain. I think that this is just an idea, and I never discussed with them about that, you know. So
honestly, I think they may have used it because they mentioned that a feature of aging is going to be like this decrease in adipogenesis as well as just lipid dystrophy with the adipose cells decreasing in size. They want to see,
and it's easy to do, also. It's a very in terms of access, it's easy. And it is easy to do, but I have some doubt about the beta-gal stains. Really, you know, since the beginning, since it was published, I have some doubts and discussed with Jan, and he mentioned me that it was done in collaboration with him. But I have some doubts. I mean that I inject tons of X-gal to some mice. I never get this kind of tissue. I mean that can you see the tissue has been collected and then they incubate the tissue in the X-gal solution. I never saw that. This is something that according to me is a kind of artifactual. Okay. What about the progenitor, the fat progenitor cells? Why, according to you, why they are turning so senescent compared to the other one? You have an idea here again? We are working on aged mice, 80 months old.
there's accumulation of this.
Well, one of the explanation is that these are cells again that are cycling, not a lot, but they are cycling cells compared to the other one that should be post-mitotic. And one of the explanations should reside in the fact that maybe these cells are more prone to develop senescence. And of course, when you treat them with AP, that works really well. If you don't have the cells, you cannot validate your experiment in the adipose tissue finally because you don't have cells that are very high and that they are not responding to the treatment, as you can see here. The other one, if they are not senescent, they are not responding to the treatment. That's something it is expected. Okay, and the last one, I don't remember what. Yeah, it was just one thing you mentioned, Hugo. I'm not agree. I disagree with the fact that I don't know if this is maybe what they say, but X-gal crystal proportion found in small perivascular cell is reduced. Because when you say that, it's exactly the same situation that we discussed just before. If you say that it's reduced, that means that the treatment is able to decrease beta-gal activity or senescence. This is not true. We are okay with that.
Yeah.
We are not decreasing senescence. We are killing the senescence. That's totally different. Okay, so I think that here okay, but whatever you are looking for, you can see that you have a strong enhancement of different parameters. Here on this slide, you can see that these guys are not experts in anything. They are looking at everything in the same time because they don't know. They are not expert in adipose tissue. I think that they are going to do something on skeletal muscle, blah, blah, blah. They are not experts. So that means that for them, what is important is that finally the different feature of aging that they can find. As mentioned by Emanuela, in adipose tissue, we know that during aging, the size of adipocyte is going to decrease. It's totally opposite to what happened with obesity. And with the treatment, you are increasing the capacity of storage of this adipocyte. Okay? So now we know. But when they did this publication, sometimes we don't know exactly what are the perspectives of such a discovery. Okay. Do you have any question or remark about this figure? because after we don't have the time to come back on that because it's a big paper. It's okay? Do you understand what they are doing? No? Farah, you don't understand what they are doing? The hypothesis that when we have senescent cell in one tissue, we have associated aging, okay, loss of function. If we kill the cells, the loss of function normally will disappear. Is that true? Is not true? I don't know. But for answering this question, they are generating a mouse model, which specifically will have the possibility to, by inducing something, to kill only the P16 positive cell. And at the end, they are just measuring, because you know this construction is on all the tissues. They are measuring what's happened after cutting them with AP molecule. They are measuring what's happened if we kill the senescent cell. As you can see, and as mentioned by Hugo, first of all, they can see that on the endothelial cells, there is less endothelial cells that are beta-gal positive, meaning that the experiment worked, at least on the endothelial cell. And on adipose tissue, they found that adipose tissue fat mass is decreased with age. You can compare the white bar. When you present, maybe next time you should show exactly what you said. The white bar with the blue one, you can see that the fat mass is decreased with aging. This is something that we know because adipose tissue, age adipose tissue has some difficulty to store. And then with AP in male and in female, you can see that you are able to reverse this effect. So to recover some capacity to store the lipids in adipose tissue, which is crucial when you are aged, etc., etc. After that, they are measuring, adipose tissue weight, adipose depot weight, the diameter of adipocyte in J and in K, which are, these are some different markers of adipose tissue, okay? So first of all, in adipose tissue and in adipose tissue, they can see that the treatment works and seems to enhance the quality of the tissue. It's okay for everyone? Okay, let's continue. Figure 2.
Okay, so this figure showcases the health span analysis of two cohorts. Basically, in the beginning, there were a mixed background of male and female mice and another mice group called the C57BL8 blood tube background. And these mice were fed twice a week with a irradiated pellet chow diet, and the C57BL6 mice with the background, they were fed, I think, 9%, and the mixed background were 5%. And as we see, over 18 months, starting at 12 months, the clearance started, and over time, when they were injected with the AP20187 and the vehicle injections, they became moribund, meaning they were close to dying. So that is what curated in this.
Is it okay for the protocol they use? So after showing that it seems to work on the tissue, we remain a protocol, and you can see, as mentioned by Samira. Okay, so they are doing this protocol. Okay, go ahead.
So with figures B and C, this is basically a log rank test, meaning that it's just a hypothesis of survival rates. And in figure B, it is shown that the survival curve has a median lifespan of 27%. As you can see in bolded, that is the control group, and then the dotted line, I don't know if you can see it as much, but that is the AP injection, I believe. And so the figure C is quite similar. There's not really a lot of differences, but it just really shows that the AP injection extended the lifespan of the mice, irrespective of the sex and genetic background.
Yes, so in figure 3, they are trying to see the effect of the cell clearance on the health span this time. So just a quick reminder between the lifespan and health span. So
We are agree on the difference about lifespan and health span? Yes? All? Okay.
So
You
I
are.
wanted to do it.
No, Bradley, they are all agree, and it's okay. We are all experts.
First, on panel 8, they are measuring the survival of the mice, and by that it means that the mice that are dying by cancer deaths only are counted. And what we can see is that with the AP treatment, there is a significant increase in both cohorts, mix and L6, meaning that the tumor latency is increased with the treatment. And you have also here two pictures of 22-month-old mice, BL6 and mixed as well. And what we can see here is that the treated mice looks healthier, as the non-treated has a different posture. You can see maybe the eye is a bit closed. She looks a bit more aged. Let's gonna say it like this. And so yeah. And finally here, so they are measuring the spontaneous activity and exploratory behavior by open field test. So on so here you have the control, and you can see a significant decrease of the spontaneous activity and exploratory behavior here as well. But these two parameters are restored when you are using the AP treatment. And this is independent of the genetic background, as we can see here with the GLC6 and mixed, and also independent of the sex.
Why they are measuring that in different strains, you know?
So
different background.
I don't know if we have an hypothesis.
Yeah, really.
It's like it's to be sure that there are no like that the genetic background doesn't interfere with the treatment or just these parameters.
Yeah, you get it. I guess this is just to be sure that the treatment is not specific of one background, okay? Because it's not because it works on black cis mice that in black cis mice that it will work on other backgrounds. So to be sure, you can do different things. You can do the same experiment on different species. That could be a gold standard, or you can at least do it maybe in different at least different background. I think that the different gender now play something that is crucial in all the study. Consider. You know why we used to consider only the male before? I mean, before 2 years ago, and today we are including, of course, female in the study. You have an idea? Why all the labs, even in Toulouse and even in my lab, we are using very often the male and not the female. Yeah, about the hormonal system, it's quite difficult to be sure that they are on the same day of the cycle, etc. So you can have some difficulty to reproduce results, but we can see that nowadays we have to, and we must, include the female, of course, because some age-related pathology should be also specific to female, and the treatment that works with men should not maybe work with female. That means that is really important. Sorry for interrupting you.
So yeah, to conclude this figure, the net and self-clearance for health plan, and move on to the figure 4. So here they are diving a little bit on vital organs, and first starting with the kidney, and specific pathology, which is glomerulosclerosis. Glomerulosclerosis, it's like when the glomeruli is a little bit atrophied and hardened, which leads to a bad filtration. And so the consequence of this is the increase of blood urea nitrogen, as you can see here, in the non-treated condition. And again, when you treat them with the AP, you can see a decrease of the blood urea nitrogen. And so it has been demonstrated here that the sclerotic glomeruli percentage is increasing in aged mice, and it's highly decreased when you put them with AP. So here you have a staining with hematoxylin and eosin to show that there is on the left panel here, what we can call glomerulosclerosis. You can see that compared to the right panel, sorry, it is more compact and yeah, what to say?
Yeah, we are not expert in vulnerability, so you know.
And here you have also a periodic acid sheet, which show a darker magenta color here, showing again the glomerulosclerosis.
Yeah.
And finally, in panel D, it was just to confirm that the senescent cell clearance was functioning in the kidney, which is the case here. You can see in 18 months, it should be nice.
Difficult to trust sometimes a picture because
Yeah,
I double check on the paper, we cannot see it very well. So.
I believe the sampling number is not really known.
Yeah. Because, well, you know that you can maybe also take advantage of that. So that in spite of low number of mice, because they have a strategy which is on off. That means that you have or you don't have the senescent cell. So they, of course, if you want to look at some maybe more precise information, you should add some animals. In that case, finally, it's quite good to have this significance with this number of mice. I discussed with them about that. I remember. I don't remember the reason why they use a number which is really weak. They are not experts in all. They are molecular biologists, really. They are not experts about everything. I mean, they never under mice and they don't know exactly what to do. But I remember that maybe they have two. I don't remember. They made a lot of issue in the animal facility, and I remember that it was difficult. And you have to keep in mind that this guy, quite difficult because this guy, you need to have them when they are old, so to keep them as old as possible, 18 months, and then they have to be sensitive to the treatment. So that means that after the treatment, you need to keep them alive. So it's quite difficult. I have a question because I think that you in the figure 2 you presented the tumorigenesis, occurrence, etc. for the cancer occurrence. Why they did that according to you?
Where?
No, why? In the figure 2, I guess it was 2. No, it's another cancer. Why do we investigate the cancer occurrence here according to you?
Well, because to measure the health span, like for me the mice are, there is an increase time before the mice can develop a tumor. So with the treatment, I guess this demonstrates that.
But what is the risk? I mean that yeah.
They check the tolerance of the treatment.
Or we can talk about, of course, maybe toxicity, but I'm not sure that AP is associated with high toxicity. The treatment itself, you mean the molecule, not the consequence of the treatment, right?
No, the consequence.
Oh, okay, so yeah, I agree. Yeah, they want to know if, as a consequence of this kind of treatment, about all we know about senescent and the control of cancer cells and aging cells, etc. They want to know if when we eliminate the senescent cell, if we don't have more, because it was the first expectation, maybe, or at least the first hypothesis, if we don't have more tumors. Because even if we know that to get tumor, we need to remove or to cancel the senescent state, even if we know that in this construction, you are not going to remove the senescent, you are going to kill the senescent cells, they have to and they must check and double check that we don't have more tumors. So that's why. You have to know that in my hand, for instance, I told you that I have the P16 knockout mice in my lab. The P16 knockout mice, when they are young, even when they are young, they are developing a lot of cancer. And precisely, you know, this is a kind of trigger because when you cross them with the KRAS, you know what is the KRAS protein? So when you cross this guy, the P16 knockout mice, with KRAS mice to KRAS positive mice, you are going to get a lot of melanoma even when they are very young. So that means that finally here, in that case, of course, this is P16 knockout mice. You are killing the P16 positive cells, but they need to be sure that the occurrence of cancer cells is not increasing. Okay. So to finish, the last one, no, this is not the last one. Yeah.
I have a question.
Sure.
Sorry.
Yeah, no, go
About
ahead.
your previous comment. Can you please get back to the previous slide?
Which one, the figure 2?
Yeah.
Okay, the cancer one, yeah, here.
Yes. So what is the optimal ratio to kill P16 plus without enhancing tumor progression?
That's a good question, but at the end, we are pretty sure that here. I have to answer. You want to make a
No, to be
answer?
honest, I have.
Well, I don't know if you already if you understood really what we mentioned just before, but these guys are not going to die with higher incidence of cancer. Okay, so that means that the treatment is not going to induce cancer. Did you get it?
Yes, I know.
Yeah, maybe, or I don't know what it is.
Yes,
So that means
I know that the
we
treatment won't induce cancer.
No. So that means that we don't have to make a kind of ratio between, the risk of developing cancer. I mean that you are reaching only the natural risk of developing cancer. Mice are going to die. All these mice are going to die from cancer, approximately all.
Yes.
Because mice are not going to die from a natural aging. They are developing a lot of tumor. At the age of 22, 25, I mean that when you open the peritoneum, you are going to find a lot of tumors on the kidney, etc., etc. But apparently, the treatment is not inducing more cancer than the natural aging. So that means that we don't care about cancer occurrence here, and we can accept to treat maybe in the future people not with AP, because you understood that you need to have the construction to get to be sensitive to that, but at least with compounds such as senolytics that should help maybe to reduce the number of senescent cells without any effect, normally any effect on cancer occurrence. Okay?
Okay, thank you.
Okay, figure where we are. The last one. Not yet. It's a big paper. Big paper.
Yes, so just here, they observed again by TEM the axial crystals, but surprisingly, they were found in the proximal tubules and not directly in the glomeruli. So here, the hypothesis lab made was that the glomerular dysfunction was due to the overactivation of the renin-angiotensin and aldosterone system. So in order to measure that, they tried to observe the angiotensin receptor 1A, and we can see that yes, it's increasing. Well, it has an increased expression in the aged mice, and when these mice are treated, the expression lower. And you also have a Western blot showing that the AP treatment is significantly decreasing the quantity of angiotensin receptor 1A. And finally, you can see here the immunolabeling of the angiotensin receptor around the glomeruli in yellow. And you can see that with the AP treatment, it's decreasing. So their theory here was that the proximal tubules were secreting senescent-associated secretory phenotype directly in the glomeruli, promoting this renin-angiotensin aldosterone system hyperactivity, leading furthermore to the glomerulosclerosis.
Easy to understand. I mean that they are still on the same line. And okay, so they have just new marker and then introduced as I really well explained by you, the different things. So here the marker is a receptor of angiotensin. It is here when you age, it's not here when you use when you kill the senescent cell. And it is associated with the better function of the kidney as we presented before.
Okay, so last but not least, they wanted to investigate the role of these senescent cells in cardiac aging. So two major features of cardiac aging include loss of cardiac stress tolerance, as well as changes in ventricular cardiomyocytes, So in these images, we have essentially the ventricular and atrial surfaces of the heart from mice age 12 months up to 18 months. And what we're able to see is that over time, specifically in the vehicle treated mice, there's increase in associated beta galactosidase activity. But with AP treatment, as you can see here, in the aortic group, that's primarily decreased. And then in B, what they did was perform transmission electron microscopy to see the incidence of beta galactosidase activity in ciliated epithelial cells and fibroblasts of the visceral pericardium, and they were able to see that those were present as well. And then moving on to loss of stress tolerance. So as I mentioned before, they were tracking beta galactosidase activity, but this time they wanted to look at the incidence of X-gal crystals. And they quantified that essentially by counting if they saw one or more X-gal crystals, it was okay if there was a percentage of that. And essentially what they were seeing was the same trend with that. With AP treatment, there was decreased incidence of those crystals. And then thankfully, to be able to prove that AP treatment was not really causing any overt or detrimental effects upon treatment, they looked at the left ventricle wall of the heart and they found no differences irrespective of the gender following treatment. And then in E, we're able to see the cardiomyocytes specifically in the pericardium. And one thing that the paper pointed out was that with cardiac aging, these ventricular myocytes are supposed to be increasing in size, but after AP treatment, there was decrease in size, proving that AP was effective in combating cardiac aging. Okay. And then last but not least, another feature of cardiac aging that I just mentioned was cardiac stress tolerance. This is typically associated with ATP sensitive potassium channels and the special subunit known as SIR2A. Over time with aging, the expression of the subunit essentially declines. So what they did was measure SIR2A expression following treatment, and we were able to see that there's recovery or maintenance of the expression of that subunit post-treatment. And then essentially what they did for time to bed, so they injected the mice with a beta emitter receptor agonist called isoproterenol. And what they did was see after it induced something called arrhythmogenesis, or just a normal heart impulse, how long did it take for the mice to die? And mice that were treated with AP essentially did not die as quickly as mice that did not have AP. And then last but not least, they also looked again at the change in left ventricle mass, as the left ventricle is indicative of how hard the heart is working to pump blood. So they saw that again with AP treatment, there was not too much change or not a drastic change in the health of that. It's really reconfirming the effect of AP.
Okay, so we had our discussion and conclusion. We categorized them into 4 areas. We saw that we concluded that for the lifespan impact, P16 INK4A positive cells, they shorten the lifespan clearance and attack might when extended in the median lifespan. As for the mechanisms, we concluded that P16 INK4A positive cells, they contribute to tumor growth and age-related decline and selective organ resistance. We also implied that targeting senescent cells could aid in age-related disease management. And then we also lastly concluded that the efficiency of the clearance, it enhances longevity, and it will also reduce age delays in the decline of mice. Our interpretation and perspectives, we see that this article demonstrated that the P16 INK4A positive senescent cells, they negatively affected the aging by promoting tumors and various age-related changes. We also saw that the clearance of these cells, like I said earlier, extend the lifespan. And lastly, we see that the cells are plausible candidates for age-related disease therapies. For the limitations in the article, we sought out that the AP treatment clearance was partial and could not clear senescent cells in the liver and the colon. We also saw that vehicle injection stress impacted the male median lifespan because they were kept being injected many times. As for future research, we would like to see reflection and optimization of senescent cell removal protocols and methods to achieve full clearance. We also would like to see investigation that would continue to observe maximum lifespan to define a more anti-aging effect. And lastly, we could further explore the utility of the P16 INK4A positive cells to in wound and
tissue regeneration. Thank you for your time.
Thank you very much. Do you have any question, any comment about the conclusion? Maybe here we are talking about the last slide presented here.
It's not about the conclusion, it's about the introduction. Sorry, can you explain me how the AP treatment targets and kills only pigs in plus
For the AP, yeah, so we didn't show this, but and before they did all of this work, they essentially tested whether spares would be possible in progeroid mice, meaning that mice that like decline rapidly with age or age rapidly. But the way for AP treatment to work is that, wish I could draw it out, but essentially imagine like a cassette, and on the right side you have the promoter region, or I can't.
We can.
Oh, can I draw it?
Yeah, perfect.
But essentially, like you have the promoter region of P16 here. That's there, and then.
No, that is not just mine because I'm turning the camera on for you. I don't know if online they can see you, but.
Afterwards, you have the, basically what's inducing the killing of the cells is apoptosis. This FK binding protein fused with caspase 8. And then afterwards, you have some like ribosomes, these things, and then EGFP so that they were able to tell apart what the resistance is. But essentially what happens is when P16 is active, there's going to be transcription of the FKP protein and then caspase 8. We can just make a start. But normally this exists as a dimer. So when it's expressed, there's a process called phosphorylation. I'm not going to explain it today on time. But essentially it'll shuffle this protein to the membrane. So bad, sorry. But it'll shuffle it from the membrane and then use dimerization so that the caspase 8 hypophotic pathway can be activated.
Okay, I need more. I have the scheme on my computer.
Yeah, I was looking
But it was really well done by I'm just going to, it's exactly what she mentioned, but if you want to be sure to be, because I think that for you, it's crucial for you to understand it. What it is. Where is that? We are done in 2 minutes. But just keep reading. Okay, maybe online you should see just the screen. That was the original paper, okay? You can see here exactly what has been. But that's good because she know it by now, So exactly the same. But to make a long story short here, as you can see here, you have the caspase 8, right? Caspase 8 is divided in different parts, and as it has been mentioned by Samuela, I guess, by Manuela, no, first by Samuela. So what you need is this molecule to make a dimerization. If in case of not dimerization, that means that the caspase is inactive. And this is mandatory if you don't want to, of course, avoid the senescent pathways that are necessary for organogenesis, etc. What we discussed before. Okay, so this is exactly how it works. You have this construction, and importantly, this construction is under the promoter of P16.
But you haven't shown that the cell targeted are activated by 16 and not.
But all the cells are going to get this construction. Okay, when you are doing that, you are going to inject the embryo, and you are going, all the cells are going to in your mice. All the cells are going to have that. Only the cells that should induce P16 will transduce this construction. And at the end, only the animals that have been treated by AP will have the activation of caspase 8, specifically in P16 positive cells. Okay? Okay? It's okay for the other one? Okay, let's continue and finish. If you have any other questions, so these are the results. But I mean that contrary to what have been presented here, they use other mice. So these mice were the mice that's called Berger-1 mice. So you know they are.
Sorry to interrupt you, but can you please rotate the camera? We can see the screen. And just ask the person who was previously presenting to stop sharing the screen. Thank you.
You will tell me if you see something, but I'm not sure we can do a 360 degrees. Wow.
Yeah, right now.
That's the best position I get. You got
Thank
it
you.
here?
Yes.
You know, in case you can, I think that that's a major paper in the field we are talking today. So that's the major paper, that's the first one. You have to know also that few years after, Darren published another story. So Darren has now his own lab, and Darren published another story specifically in brain with this kind of mice, specifically in okay? And they say that, of course, when you target senescent cell in brain, you are going to delay the appearance of the Alzheimer's disease, etc., etc. So as a proof of concept, it works really well. But now we have to transpose that for human. Here, this is the construction done in mice, but it needs this construction. You have to understand that you cannot do. It's not because you are going to inject AP to human that will work. The human they don't have the construction, right? Okay, and the result we are not going to explain today, but you know the results are approximately the same that have been presented by Hugo. This is the skeletal muscle, for instance. This is the adipose tissue here, but we have the same result here. It was just because they use not natural aging mice. They use Berger-1 mice, which are a model of mice that age very fast. Okay. Do we have to? Yeah, maybe we have to reshare also for everyone. Do we have any question? Any other question about this paper? Fantastic paper, of course. I have some questions that are maybe more technical than. Okay, can you explain the GFP is done? We have a higher GFP again. Attack you explain because we come back. The cancer is done also. Do you have any extra question? Any comment? Yeah. In English? English for Emanuela and everyone. Oh, yeah, good. No, English. Yeah, the good English. What do you want to say?
Sorry. The strategy of attack is before or after the other strategy of Sultigen with Vidanciclovir?
That's a good question, but it's not for me, for you, right? No, not in this one. But you know, with ganciclovir, of course, this is this paper. If I got it correctly, this paper was before, of course, all the strategy that was able to kill the senescent cell by ganciclovir. You have also navitoclax, etc. All the senolytic reside on this kind of discovery. So this one, I'm not sure, because this one, if you remember, 2016, and the previous one, 2015. Two nature paper in 2 years. Thanks to this mice model, you know. And I meet them. It was something like 2018, and that's why Darren has now his own lab. So if you're interested, of course, to make a kind of a training in Minnesota, I don't know if it's good or not. Yeah, very good. So, but you can tell me, and of course, Darren or Jon or at least maybe James Kirkland also, who is interested by better understanding the senescence of adipose tissue, etc. in this model. And I have the opportunity also to get this model in my animal facility, but finally we didn't do that, so we opted for another solution. But this is a fantastic team. They are very nice, very open-minded, and maybe you have some other question in the future that could develop in this kind of lab. Okay. Other things before finishing. Okay, so we are done. We did 3 different workshops. Keep in mind that every workshop is in terms of different questions we will have in the exam should take note, etc. And the next one should be for now, but maybe it could change. On November the 14th could change because a lot of people told me that they have also other things. But I'm going to try to sit and find maybe another date where you guys are all available. Thank you very much. Thank you for your talk. Thank you very much.
Just a question, please. When will the exam will have state?
Going to ask you, I don't know we can do that, maybe to put it in the Google Drive or what. All the presentations that have been made, we are going to collect, I'm going to collect them, all of them, to recheck them to see if there are no major mistakes. And after that, to put them, I don't know, on the Google website or whatever, okay? But don't send, if you send it by email, I think that is going to be too big sometimes. Oh, we're gonna try. So
I just have a question. When the exam will have points?
You have a question, Yamina?
Yes.
Sorry.
When we will have the written exam?
Yeah, it will be a written exam. Yeah, 2 versions in English or in French. When? Oh, in December, around I don't know, before Christmas. I don't know exactly. I will let you know. I will send you an email to tell you where we are.
Thank you.
Thank you very much. See you.